Good morning, everybody, and welcome to day 2 of the Variability, How to Deal with It, Interpret It, and Learn from It meeting. We've got an excellent day lined up. The first session will be to do with clinical settings, the second session, industrial sessions areas, and then the third will be the statistics and how to deal with variability. So without any further ado, I'm going to introduce the chairman of the first session, who is Professor Hugh Montgomery. Hugh is a consultant intensivist and director of research at the Whittington Hospital. He's head of the Centre for Human Health and Performance at University College London Division of Medicine. Over to you, Hugh.
Well, thanks very much indeed, Mike, and welcome everyone to this stellar symposium, I think. Some quick reminders. If you want to share thoughts on social media, the hashtag is #variability 2020, so 2020. If you want to submit some questions as they occur to you, you'll find a Q&A button at the bottom there. So feel free to post them. You can do that anonymously if you tick the box, but otherwise you can welcome to put your name and institution. And we'll try to answer as many as we can. We may not get to them all at the end. And finally, just a reminder, there's going to be a networking session at the end, and you'll get a link to that post at the end. And there may be breakout rooms for up to 5 people to allow you to discuss in a little bit more detail. So this morning's session is about clinical setting variability. So what about biology? What about patients? What can we learn by the study of variability? And I'm joined by some stellar panelists, I have to say. I'll introduce them briefly now so we can save time as we go along. First up will be Lindsay Edwards, who, apart from being pop star, record producer, physiologist, cyclist, racing car driver, is also director of machine learning and artificial intelligence at the drug company AstraZeneca. We're going to be followed by Clare Frobisher from the Institute of Applied Health Research at the University of Birmingham, where she's one of the lead team for the British Childhood Cancer Survivor Study. That's the first study of its kind, looking at cancer survivorship in children, studying nearly 18,000 individuals. Next up is my friend and colleague, Professor Dan Martin, now at the University of Plymouth, who's a fine clinician, but also a fine physiologist, who's led a great deal of work on Everest and in other places, looking at variability in human adaptation and response to hypoxia. And finally, you have to put up with me talking for 10 minutes briefly about COVID. So first up then, Lindsay, tell us why you think variability is important and why it's a feature, not a bug.
Good morning, Hugh. Good morning, everybody. First of all, huge thanks to Mike for the invite to come and speak to you today. It's a real treat to be part of a Physiological Society event again. A quick background on me. So I'm a vice president at AstraZeneca and the head of artificial intelligence for respiratory and immunology. Before that, I was the VP of machine learning engineering at GlaxoSmithKline, and before that, I was an academic at King's College London. And the reason it's a treat to be in a PhysOc event again is because I have my PhDs in physiology, and I have in the past been a proud member of the Physiological Society. So it's great to be here. So hopefully, well, so the first thing is we were talking about outliers before we came online, and I'm definitely an outlier. And I think probably what I'll be talking about today will be very different to some of the things that have been discussed before. I hope so. And that's sort of partly because I've come to view variability in a very different way, by necessity, really. So I think part of the motivation of this conference was that we tend to think about variability as being something of a bad thing statistically, because it sort of too much variability tends to damage your statistical power, or it can make it difficult to interpret your data. And so this has led us as experimental scientists to move towards weird systems like clonal cells and clonal animals. I spent, I guess, the better part of the last 15 years modeling biological systems in various different ways. And for me, variability has become essential. So what I'd like to do is walk you through that journey a little bit and some of those experiences. This is quite mathy, so I'll sort of apologize beforehand, but I will try and stick around. And if there's, first of all, I'll try and be as clear as I can. And if I'm not, then that's on me. But I'll also sort of try and stick around if there are any sort of questions afterwards. So Rosie, could we go to the next slide, please? So let's just look at different ways of modeling biological systems. And just at the highest level, remember that we all model these systems all the time, whether it's your kind of conceptual idea of what you think is going on in a complex biological system, whether it's a figure, some sort of drawn figure, whether it's a statistical model, or whether it's a machine learning model or a mechanistic model. What we're really doing is we're trying to say, I have some idea about the way I think the world is. You know, to paraphrase George Box, that idea is wrong, but it might be sort of useful in some way. And what my experiments are doing is attempting to either validate the way that I think the world is or challenge the way that I think the world is. And that's sort of broadly speaking what we all do. What I put down here is kind of 3 different ways of doing this experimentally. So the first one is the way that I was taught at university, the hypothesis answering approach. And so as a biologist, you take a biological system and you inject some controlled variability. You change the system with some insult or intervention, and then you sort of gather data before and after you've done it, and you do other stuff to control for other things. But this is the basic setup. So there are huge positives to this. The first positive is that if you design your experiment well, you should answer the question that you've gone in to answer. So that's good. The downside is that very often these data sets, and I know this from bitter experience, don't play well afterwards because they've not been designed with the intent that they would be reused. And because the variability has been hopefully well controlled, apart from the variability that you've injected, it's an extremely biased data set. And also we have a tendency in biological sciences to measure things in arbitrary units because we're only really bothered whether something changes, not necessarily in comparing sort of stuff between experiments. So let's look at a couple of ways of modeling systems. So the sort of classical systems biology approach is that you take a system and you perturb the living daylights out of it. So you hammer it with compounds and genetic knockouts and knock-ins and knock-ups. And the idea is that you probe every possible cell state. Now, if you're able to do that, if you were able to explore every possible state of the cell and build a model that captures all of that, you have effectively kind of finished doing experiments. So I once heard Bernard Paulson make this quite provocative but interesting statement that a gene, you know, that a genome well sequenced means that you've changed it from an experimental problem to a computational problem, because all of the information about the actual sequence is now on your hard drive. Again, by the way, I'll leave that there as a sort of talking point. I'm not saying I necessarily agree with it, but the general idea is that if you probe every possible state of the system, you've captured it all and you've got a model, and then you ask questions of your model. Of course, the downside of this is that it's rarely possible to really do that in practice. You can only probe a small subset of possible states of the cell or the system, unless it's a very simple system. The last one is you constrain your model. And this is something called inductive bias. So we'll come back to this quite a lot in this talk, but inductive bias is really a way of saying, I'm not going to kind of leave all the doors open. I'm going to put some prior knowledge on my model and assume that certain things are true. Then I'll need a lot fewer data to kind of explore that space of possible models. Of course, the downside, so the pro of this is that it's easier to get the data and you can limit the system you're interested in. So it could simply be a simple enzyme within a system. But the downside, of course, is that your assumptions can be wrong. And if your assumptions are wrong, you can get yourself into all sorts of difficulties. So next slide, please, Rosie. So I quickly just want to talk to you about this idea of thinking of cell state as a vector. And the reason I want to talk about this is because it's just a kind of getting you to make this hop between biology and then the kind of mathematical representation of that biology. So imagine for a second that you have a simple cell where you can measure every single property. Let's say it's so simple that it only has 2 variables that you need to measure and you're done. Now, if you think about it, you can then plot every possible combination of those 2 variables on a simple XY axis. You know, so 0, 0 would be down here and 100, 100, and your 2 variables would be up here. Now, here's the trick. You can expand that out sort of pretty much to an infinite space. But let's say you've got a slightly more complex system. You've got a set of measurements, let's say kind of n measurements. There are 100 measurements that completely define that cell. And now you can think about the states of that cell as being a point in a 100-dimensional space. You don't really have to imagine a 100-dimensional space. Most of us do it in 3-dimensional space, but that's kind of fine. So the next thing that's worth noting is that actually not all of those points can exist. So if you're a cell, there's actually really quite a limited number of possible combinations of values that are true. So rather than existing in this kind of 100-dimensional space, the cell exists in a sort of in a subspace of that 100-dimensional space. And a nice way to think about this mathematically is a thing called a manifold. And manifolds mathematically you can think of as being a surface, but it's basically a subset of the whole space in which the cell can exist. And so one of the points I want to kind of raise today is that you can think of a cell state as being expressed as coordinates on this manifold. So if you think this manifold as a surface that captures every possible state of the cell, then every possible sort of state the cell can be in is a point on that surface. And our goal as scientists is to either learn as much of that manifold as possible. So that's the systems biology approach of just peppering the cell with everything and kind of shining a torch on all the different places where the cell can live. Or if you're an experimentalist, it's kind of you've got a cloud of points on the manifold here, and you do some stuff, and you're a cloud of points on the manifold here. But effectively, it's just a way of conceptualizing in your head this business of modeling cell. So if you could just quickly go forward a slide, Rosie. I just want to give you a kind of very simple example. So let's take this as a cell, okay? So in the cell, we've got a couple of importers. One imports this thing called A, one imports this thing called B. We've got this enzyme called an AB ligase that joins the A and B together and creates AB. And you've got an AB exporter that shuttles these things outside of the cell. So taken at face value, the importers, the exporter, and the ligase could all have kind of any value you like. AB concentration could be anything you like, but in reality, we all know that's not true. So if you could just click, Rosie, there are some simple constraints. So first of all, the importers and the exporters will live in a world where they're physically constrained to the rates that they can have. Next click, please, Rosie. We can assume some sort of steady state. So if A, B, or AB were allowed to accumulate infinitely in the cell would explode. So over a period of time, the rates of these things have to match. And then last click, please, Rosie. We could also posit something called an AB sensitive function, some function or functions in the cell that require AB to be in quite a tight range. And already you've taken this kind of all possible values thing, and you shrunk it down to the space in which the cell can exist. And if you're troubled by AB sensitive function, think about pH sensitive enzymes that will only function in a tight range of pH. Next slide, please, Rosie. Okay, so you've got this subspace of possible values in which a cell can exist. And this is the sort of intuition for a manifold. So in a 3-dimensional space, let's say these are 3 measurements in the cell, actually the cell can only operate on this surface. And then every kind of thing that the cell can do is a point on this surface. And so here, this is just a completely made up example, but you could say, for example, that a dividing cell would be this point at the top, and maybe the z-axis is metabolic rate. This thing on the bottom is a kind of quiescent cell. But the question here is, how do we learn this manifold? So next slide, please, Rosie. So let's tackle this idea of inductive bias. So let's take an example where our inductive bias completely defines the model that we use. In other words, no data are required at all. And this would be what we would think of as a mechanistic model. So this is a mechanistic model of a mitochondrion. And what's happened here is we've just said, look, we know how all this stuff works already. We know how enzymes work. We think we know what the parameters are. We're going to plug all these things together. We can run simulations, but there's no data required after the model is built. And that's because we've entirely encoded our assumptions, our prior, if you want to be a Bayesian about this, we've entirely encoded our prior information in a way that doesn't require, or we're not going to ask for any data to modify that at all. So this is kind of full inductive bias, no data required. Next slide, please, Rosie. So the kind of next stop on our trip would be something like this. So this is a population pharmacokinetics model. So in this model, and this is the closest to a kind of a traditional statistical approach, if you like. So a general linear model would fit into this framework. But what we're saying here is this is something like population pharmacokinetics. So what we've said here is we have some prior knowledge about the nature of the functional form that relates the concentration of a drug to time. And so that's this function of time here in the equation that I put here. And then in this version of the equation, we've assumed that the noise is normally distributed. And what you've done there is you've kind of massively shrunk the space of all possible models. You're not saying anything goes. You're saying, I believe that there is some functional form to this model, but then you're using data to fit that model. And you would use something like maximum likelihood or least squares estimation to kind of find the statistically best fit for the model that you've come up with. Okay, next slide, please, Rosie. So let's start to reduce inductive bias a bit more. So these are network models, and this is where variability becomes essential to learning the model. So network models effectively say, well, my inductive bias here, my reasoning is that things are connected to other things, and I need a way of estimating the degree to which things are connected to other things. But beyond that, I'm not going to make too many assumptions. Now, one of the most kind of well-worn techniques for learning these models is something called WGCNA. Lots of other techniques are available. But WGCNA it stands for Weighted Gene Co-Expression Network Analysis. Basically, what it does is it takes a lot of, say, transcriptomics data, and it looks for relationships, pairwise relationships between transcripts. So you would calculate the pairwise correlation matrix, or use a thing from information theory called mutual information, which is a bit more robust to nonlinear relations. But you sort of learn relationships between transcripts, and then you build a network that says, well, if these two transcripts correlate, I'm going to draw a strong edge between them, and if they don't correlate, I won't. So the key thing here is that variability is absolutely required. You require a spread of data in order to allow you to detect those correlation structures, to detect those mutual information patterns. And actually, if you have data that's too constrained, where variability is too low, it's very, very difficult to learn these network models. Okay, next one, please, Rosie. So on to inductive bias 4, and this is one of my personal favorites at the moment. So this is a causal DAG, and DAG stands for a directed acyclic graph. This is super trendy at the moment. I mean, so causal reasoning and causal inference in statistics has been with us for quite a while. But the sort of impact of causal statistics in machine learning is very exciting at the moment, and it's kind of the bleeding edge in terms of what's happening in modern machine learning. So very quickly, this thing on the left is a directed acyclic graph. It's a causal directed acyclic graph, and all that means is that I'm making causal, my graph encodes some theories of causality. So again, this is my prior, or this is my inductive bias. So in this case, what we're saying is we've got the simple system where time of year causes, has some causal effect on the probability of both rain and whether or not your sprinkler is on in the garden. Both of those things affect whether you might have a wet pavement, and that in turn affects whether you have an accident. You might have an accident. So there are a couple of properties of causal DAGs that are important. So one of them is this thing called the global Markov property, and this is a property of the graph. And very simply, the global Markov property says that every d-separation in the graph, and I'll explain what that is in a second, maps to an independent statement in the data. So d-separation is kind of like independence on a graph. So if you look at A here, then all of the information from these nodes T, R, and S comes through W to get to A. So A is independent of T and R and S conditioned on W. What you're saying is that if you condition on this variable W, you effectively factor out all the possible information that lies above it. And so this is saying that there's some property of the graph, this d-separation property of the graph. And the global Markov property says that if the DAG is representative of the data, then the data should reflect that in an independence relationship. So you should find that if you control for W in your data, then A should be independent of T and R and S. Now, of course, independence implies dependence, and dependence is only there really if there is some variability in the data for you to detect that. So you're better with variability and better able to detect dependence and independence if you have variability in your data. And faithfulness is kind of like this stood on its head. So faithfulness says the same thing. It says if you detect an independence in your data, so if you discover, for example, that W is independent of T conditioned on both R and S, and it should be if the causal DAG is correct. So that's faithfulness. And in both cases, you require variability in your data in order to make these both dependence and independent statement. So my point here is that variability becomes increasingly important the more you reduce your inductive bias. One more slide, please, Rosie. Very quickly, there's something fascinating about causal structure learning, and that's this idea that sometimes bias between your data sets, which I mentioned briefly at the top, is good. I know I'm a little time constrained, so I'll try and be quick. One of the most common problems that I encounter when I'm trying to join data sets that have been acquired for a different purpose is that there's a huge bias between them. So effectively, if you took these two data sets, joined them together, did something like principal component analysis and looked at the first two components, you would have a blob here and a blob here, and this is experiment 1 and this is experiment 2. If you just flick forward a slide, Rosie. So there's good reason to believe that this, rather than being a very troublesome thing, could turn out to be a very useful thing. And it's this idea from the causal literature called the principle of independent mechanisms. And very quickly, the principle of independent mechanisms is that when you look at a causal mechanism, you're looking at something in the real physical world. And the example I use here is the relationship between altitude and temperature. Now, there are two different ways to view the correlation between the altitude of a city and how cold it is. One of them is that temperature causes altitude, and one of them is that altitude causes temperature. We'll leave aside the idea of confounding just for a second. So in the one case, you could intervene on the temperature. So you would raise the temperature and your city would sort of sink. And the other one is that you raise your city and you would find that the temperature changed. Now, we knew that, that second thing is true. And actually, what we assume is because there is a physical mechanism that relates altitude to temperature, that the functional form of that relationship will be robust to stuff going on around the outside of it. And this is called the principle of independent mechanisms. We say that the distribution of T given A is independent of the probability of A. Now, this isn't the same as saying the data are independent. This is that the distributions are independent. It's a subtle point that we could perhaps touch on later. But the main thing here is that this is a real thing in the real world, and it should be robust to the stuff that goes on around it. And the key thing here is that when we have this bias in data sets, it's becoming clear that that bias can be used as a method for detecting whether you have a causal relationship that's real or a causal relationship that's not. So just flick forward one slide, Rosie. Oh, sorry, one more. This paper was at ICML this year. The ICML is one of the, it's like the nature of machine learning. Two of the names here on here, Yoshua Bengio and Bernard Scholkopf, for two of the biggest names in machine learning. Scholkopf invented support vector machines. Bengio won the Turing Prize last year. So, this is a huge area at the moment for machine learning. So very briefly before I close, one more slide, Rosie. It's difficult not to talk about neural networks at the moment. One of the reasons why neural networks are so exciting for a lot of people is because they are universal function approximators. So this means that a fully connected neural net like this one can learn any function of arbitrary complexity as long as you have enough layers, which is the kind of kicker here. But there is a flip side to this, which is a fully connected neural net has no inductive bias. If you can learn any functional form, then there is nothing off the table. And this is one of the reasons why they're so data hungry. They're incredibly difficult to train, and actually why the most successful applications of neural nets, things like convolutional neural nets or transformers, have an inductive bias built into them. And again, this is a kind of active area of research in the machine learning community, which is sort of how do you increase inductive bias and sort of shrink the amount of data that you need to train the model. So very quickly to summarize, one last slide, please, Rosie. So when we sort of think about a traditional experimental design, we're both introducing controlled variability, but also controlling the variability that we don't want to see. However, many modern machine learning techniques actually require variability, and the lower your inductive bias, the more that variability is required for you to be able to learn the model that you need to learn. And finally, recent research in causal DAGs and causal machine learning suggests that one of the most troublesome sources of variability, the bias between the studies that we run, might turn out to be actually rather useful. And hence the sort of title of my talk, it's a feature, not a bug. Okay, I hope you stayed with me. Thank you, everybody. Thank you for the invite. And I'm going to sit back and listen to what everyone else has got to say.
Fabulous, Lindsay, and a great start. So a really good meaty start as well about why we should be considering variability as a tool to explore systems. Thanks very much, Lindsay. We'll move on. We've started a little late, but let's see if we can catch up a little on time. It's my pleasure to introduce Clare Frobisher. I mentioned her earlier on, lead on the first and therefore very large study of cancer outcomes in children. And she's going to tell us about heterogeneity in mortality in cancer among all refinery and petroleum distribution workers. Claire, over to you.
Okay, hello. So yes, thank you for inviting me here today to talk about a cohort that we've been investigating. And it's within UK oil refinery and petroleum distribution workers. Could I have the next slide, please, Rosie? So what the cohort involved was a male cohort of workers who were originally employed within the oil refinery and petroleum distribution industry, and they were employed from 1946 up to 1974, and they had some employment after 1951. So the cohort is split up into those that were employed at 8 UK oil refineries, so this equated to about 28,500 male workers. And also we had workers from 476 UK petroleum distribution centres. And again, this was about 16,500 workers. So the objectives of the whole cohort was to look at whether there was any excesses or deficits in mortality and cancer incidence in relation to the workers compared to the general population, and to determine whether any of this experience might be related to occupational exposures. So what I'm just going to concentrate on here today is to just give you a little snapshot of where we found some significant excesses in the mortality and in the cancer morbidity, and how this varied across particular factors which we're investigating within the cohort. Could I have the next slide, please, Rosie? So first off, I want to look at the mortality analysis. So to do the mortality analysis, each worker entered the period of follow-up at the end of a minimum of 12 months of employment, or 1st of January 1951, whichever was the later date. And then we followed them up to either the end of the study period, which was the end of 2016, their date of death, or the date when they emigrated, whichever was the earlier date. So to look at mortality analysis and to see whether the workers were experiencing any excesses in mortality, what we looked at was standard mortality ratios, which are SMR, abbreviated on this slide. So to calculate SMRs, we're looking at observed number of deaths over expected number of deaths. And this was done for all causes of death, and then for specific non-neoplastic and then neoplastic causes of death. So where we were looking for excesses, the SMRs and also the 95% confidence intervals for the SMRs would all be above 1. So if there was an excess compared to the general population, the SMR and the 95% confidence intervals would all be a value of greater than 1. So when we found a significant excess of mortality in the workers, we've further explored within these particular factors shown on this slide. So we looked at year of death, period from commencing employment, decade of hire, and job type. And what we looked at, we looked to see if there was any significant heterogeneity within these factors for the SMRs. And then also we did a multivariable Poisson regression model, which produced adjusted relative risks. So in the model, we took account of all these factors plus attained age. And what we got from the Poisson regression models is adjusted relative risks, which could be interpreted as ratios of the SMRs accounting for these factors. So next slide, please, Rosie. So looking at the deaths within the oil refinery workers, the only non-neoplastic death that was in excess in the oil refinery workers was seen for asbestosis. And here we see that the workers had a threefold increased risk compared to expected for this particular type of death. And for the neoplastic causes of death, the only significant excess was seen for mesothelioma within the workers, and the workers were experiencing over a twofold excess risk compared to what you would expect from the general population. So then we looked at the variation within these particular causes of death by year of death, by period from commencing employment, by decade of hire and job type, to see if there's any significant heterogeneity for these particular causes of death. Next slide, please. So the only significant factor where we found significant heterogeneity was for mesothelioma, and that was by job type. So on this slide here, I presented the standard mortality ratio by the particular job types within the oil refinery workers, and I've also presented the relative risks as well from the multivariable Poisson regression models. So if we look first at the ratios, the standard mortality ratios, the highest compared to the general population, the highest excess was seen in the riggers. There was seen a nearly a 6-fold excess compared to what was expected. And also there was excesses over 4 times for pipe fitters at 4.2, and near to that was also the other craftsmen at 3.9. Engineers had a 3.45-fold excess risk, and the fitters also had a 3-fold excess risk for mesothelioma compared to expected. We also then did a Poisson regression, and we used the admin and the clerical workers as a baseline group. So the highest excess risk for mesothelioma compared to the admin and clerical workers was seen in the riggers. They had a 6-fold increased risk for this particular mortality type compared to the admin and clerical workers. And also the pipe fitters and the other craftsmen had a increased risk of about 5-fold compared to the admin staff. Okay, next slide, please. So then what we're looking at is what's happening within the petroleum distribution workers, and the only cause of death that was in excess within these workers was for prostate cancer. The SMR here was a 1.1 with a 95% confidence interval above 1 as well. We also looked at the heterogeneity across the factors again, across year of death, period from commencing employment, decade of hire and job type, and there was no significant heterogeneity in all these factors for prostate cancer. Next slide, please. So what we did next is then we also looked at the cancer incidence within the workers. So what we did in the cancer incidence analysis is that each worker entered the follow-up at the end of 12 months employment or 1st of January 1971. There's a typo error on this slide. It should be 1st of January 1971. So it was either after their 12 months of employment or after 1st of January 1971, whichever was the later date. And the workers left the period of follow-up at the end of the study, 2015, date of cancer registration, or whether they emigrated. And we calculated SIRs, which are basically observed number of cancers over expected number of cancers. And this was done for all cancers and then specific cancer sites. And then we looked at heterogeneity again for those that were in excess for year of registration, period from commencing employment, and decade of hire and job type. And this was done by looking at the variation in the SIRs and also from the relative risk from a multivariable Poisson regression model. Next slide, please, Rosie. So in the oil refinery workers, the excess for the particular cancer sites was seen for non-melanoma skin cancer at 1.2-fold increased risk and for mesothelioma at 2.4. And then we looked at to see whether there's any significant heterogeneity by the key factors again. Next slide, please, Rosie. And the only significant heterogeneity that we saw was for mesothelioma, again by job type within the oil refinery workers. And so if we just really concentrate on the relative risks on this slide, the highest relative risk within these particular workers was seen for the fitters and for the other craftsmen at 4.8. And there's also some other ones that had an increased risk of 4 times that of the admin and clerical staff, and that was for the pipe fitters, the riggers, and close to that was the engineers at 3.8. Next slide, please. Thank you. So for the petroleum workers, the excess risks that we saw was in lip, non-melanoma skin cancer, connective tissue and bladder. And next slide, please, Rosie. The only significant heterogeneity that we saw for the non-melanoma skin cancers within the petroleum distribution workers was for job type. And if we just concentrate on the relative risks, the ones that were over 1 was for the drivers at 1.1 and 1.3. I mean, the confidence intervals don't suggest that's statistically significant, but they are very close to being over 1. So there was potential evidence that there could be an increased risk for non-melanoma skin cancer in drivers and in the managerial job types in the petroleum distribution workers. Last slide, please, Rosie. So in summary then, among the oil refinery workers, the excess of mesothelial deaths and cancers demonstrated significant heterogeneity by job type. And with the petroleum distribution workers, the excess of non-melanoma skin cancers demonstrated significant heterogeneity in the relative risk by job type. Okay, so that concludes my talk for today. Just look at the last slide, Rosie, thank you. So thank you for inviting me here today, and just to acknowledge this was funded by the Energy Institute.
Thank you very much indeed, Claire. Moving swiftly on then, and Dan, if I could ask you to, if you can hold to time, that'd be most helpful. Dan Martin, professor in Plymouth, a mountaineer himself, and quite a good example of variability in that Dan performs quite extraordinarily at very high altitude when many of us mere mortals do not. Dan, tell us a little bit about adaptation to hypoxia and the variability therein.
Thank you very much, Hugh, and thanks everyone for tuning in and also for the invitation to speak here today. We are running a bit behind time, and I shall try not to exacerbate that. Over the next 10 minutes or so, I really just want to highlight some unusual outliers in the animal kingdom that demonstrate variability to hypoxia. And I'm going to do that through a sort of species, then race, then individual process. And this is going to be pretty light on data on purpose. Next slide, please. Just a very quick reminder to you, just before we get going. Next slide, please. Life began on Earth around 3.5 million years ago. Next slide, please. And in that environment, there was virtually no oxygen whatsoever. Oxygen only came about following the evolution of photosynthesis, where cyanobacteria released oxygen, this potentially toxic molecule, into the Earth's atmosphere. Next slide, please. Some bacteria and archaea were able to use this oxygen to generate energy in a process we now call oxidative phosphorylation, and through a process of endosymbiosis, these then became the mitochondria that we have known to love and led to the creation of multicellular life on Earth. Next slide, please. So, the primary oxygen sensing mechanism in cells, as you all know, is the HIF1 alpha pathway, and virtually every eumetazoan on Earth expresses HIF1 alpha, prolyl hydroxylase, and von Hippel-Lindau protein, just demonstrating how important the process of sensing oxygen is to life. Next slide, please. And this really just shows you why there might be so much variability in adaptation and response to hypoxia. The HIF1 alpha complex, once activated, upregulates many different genes across the genomes via HIF regulating elements. And variability in the expression of those genes perhaps describes some of the variability in phenotypes that we see amongst different species. Next slide, please. So I'm going to just start, and the first of those sort of three categorizations, I'm just going to look at a few outliers, 3 outliers actually, in the animal kingdom, just to show you how interesting outliers are between species. Next slide, please. The first of my outliers is a mammal. It's the blind subterranean mole. The blind subterranean mole, as you can see, it has no eyes and lives underground for its entire life. It lives in burrows, and the oxygen concentration in those burrows can get down as low as 7% oxygen, so incredibly low levels of oxygen. Next slide, please. This mole has developed a whole host of different methods to tolerate low oxygen. I'm not going to go through these in detail, but you can see some of them quite closely related to those upregulated processes from the HIF1 alpha pathways, such as increased VEGF activity and increases in capillarity in various tissue beds. Next slide, please. Our next outlier is a bird, the bar-headed goose. These geese live primarily along Mongolia and the Tibetan high plateaus in the summer, and then they fly south over the Himalayas down to southern India, where they rear their young and then fly back over the Himalayas at the end of the season. So unlike the mole, which could remain fairly sedentary at times if it wishes to, flight is the most energy consuming locomotive process in the animal kingdom. Yet this bird can soar over the Himalayas with great ease. Next slide, please. The bar-headed goose has an array of different adaptive phenotypes that you can see within it. And again, I'm not going to go into these in detail due to time, but here you can see, because it needs such an enormous amount of oxygen to fly, changes in its ventilation, particularly in minute ventilation and lung volume, compared with similar birds who are not exposed to such high altitude. And this much greater affinity of hemoglobin to bind oxygen, and in a very temperature sensitive manner. Next slide, please. The third of our examples in the animal kingdom is what you might call an extreme outlier. The Crucian carp has the ability to withstand anoxia for prolonged periods of time in freezing lakes over winter. Next slide, please. It has developed a whole host of processes by which it can withstand anoxia. Not only it too, like the bar-headed goose, has this great affinity for hemoglobin's great affinity for oxygen. It can change the morphology of its gills to increase the surface area of the gills. Importantly, it can downregulate some of the highly energy consuming processes in the body, the main one of which is protein synthesis. But the clue to its real ability is that it has perhaps the largest glycogen store of any of the vertebrates ever studied. Next slide, please. It is an extreme outlier because the Crucian carp can metabolize pyruvate from the sort of glucose and glycogen stores, and also from lactate. And what it does, it uses pyruvate dehydrogenase in the mitochondria to convert this into acetaldehyde, and then via alcohol dehydrogenase to ethanol. So what it does is this very inefficient process, losing high energy carbon compounds, but what it does is to avoid the production of lactate by this fish, so that it can maintain a completely anaerobic respiration during the winter months underwater. Next slide, please. So I'm now just going to move on to some within species differences, so differences within races, and we're going to look at humans here. Next slide, please. So humans developed, probably evolved in the sort of eastern area of Africa. From there, humans spread out across the entire planet. But some of these races spread out to areas of high altitude, and you can see them marked in red on the diagram on the right, highlighting the three key high altitude populations that we currently study today: those from East Africa, the Sherpas from Tibet, and those people coming from South America. Next slide, please. There are really incredible differences between these three key populations at altitude, which I'm not going to go into in detail at all here. But what you see is a sort of spectrum from the Tibetans, who have perhaps been at altitude for the longest and the highest. They display an increased resting ventilation, they have lower oxygen saturation and lower hemoglobin concentrations compared to the other populations. At the other end of the spectrum are the Ethiopians, who, although they live at altitude, physiologically look extremely similar to those of us who live at low altitude. And the Andeans sit somewhere in between these. So the variability expressed by these populations show us important things about how humans have adapted to hypoxia over time. Next slide, please. When you look at the genetics of these three populations, you can see great similarities, but also perhaps more interesting, some really distinct differences in the way that their genomes have developed in these separate geographical areas, leading to these different phenotypic adaptive responses to high altitude. Next slide, please. And really the crux of what I want to talk about today is in individual variability, the variability that we see between people when we conduct experiments in similar environments. Next slide, please. It's really important as a physician for me to understand the differences between individuals, and Hugh is going to discuss this further in the next talk. Very close links between what we have learnt perhaps at high altitude and the hypoxic experiences that we see in our patients who have suffered from COVID over these recent months. What I want to know as not only a clinician but as a physiologist is what determines the difference between these two groups of outliers, the person who needs a helicopter evacuation because they're so hypoxic at about 4,000 meters, and then the group of individuals who successfully stride to the summit of Mount Everest at 8,848 meters. What is it that determines these two outlying phenotypes? Next slide, please. The only real data I want to show you today is just 4 pieces of data, just to outline my point here, and then I shall close. Next slide, please. Part of my PhD project back in 2007 was to collect arterial blood gases from people, including myself, just below the summit of Mount Everest on their descent. These are the data which many of you may have seen before. Next slide, please. What everyone focuses on in these data are the average and standard deviation of the data. And most interestingly, next slide, please, when you look at that data, you see amongst the 4 subjects for which we took blood gases from near the summit of Everest, you've got 2 quite low values and 2 higher values. The dreaded reviewer 2, when we submitted this manuscript for review in New England Journal of Medicine, one of their comments was that they could not believe the data that we had presented, that clearly something must have gone wrong during our experiment due to the high degree of variability of those data, and that they would expect these values to be almost identical in each subject. Next slide, please. A few days after we published these data, an editorial was published in The Lancet by John West, and he too described, and I'll just read this out in this final slide now, that the great variability in the data is difficult to explain, but hardly detracts from the extraordinary achievements of obtaining arterial blood gases under these arduous conditions. So John West too, implying that there's something wrong with these data because there is variability. And I would like to finish by saying the beauty of these data is their variability. Thank you very much.
Dan, that's wonderful. And I strongly encourage you to look at anything else Dan has online about his data, because in fact, this research has explained the variability, which gives us a way of understanding the biology of all human hypoxic adaptation. Right, it's down to me now to move on. So Rosie, if you'd like to click next, please. I'm going to be talking about COVID disease and variability. Next slide, please. My grandmother was wise, all old people seem to be, and she always said eggs are eggs, and eggs are all the same sort of thing. And she's true that's true if you're saying that eggs are female reproductive cells. But to ignore the variability is crazy. So here we have a chicken egg versus an ostrich egg. Next clip, please. There'll be a test at this end. That's obviously frog. Next one again. This is the stink bug egg. Next one again, followed by octopus, and a click again will be skate, and the click again is horn shark. So whilst these are all female reproductive cells, one couldn't say in any way that they're the same. Next slide, please. And furthermore, the chickens that lay them are different sorts of chickens in different sorts of places. Next slide, please. Even if we're just dealing with chicken eggs. And also we can have them as free range or organic. Next slide, please. And finally, of course, we could prepare those eggs in a variety of different ways. And if you click again, and then again, please, Rosie. So it is not sufficient, and I think you've learned that from this morning, to just label something as being one thing and therefore that everything must be identical. You have to consider variability to try to understand the differences. Next slide, please. Now, this is the SARS-CoV-2 virus. It's a coronavirus, and you might have read from the press or thought from the press that this is just one virus that hits you and it makes you sick, or it doesn't, and that what you get is COVID-19 disease. So just basically one disease. And this just isn't true. Next slide, please. And again, please. So firstly, whilst it is indeed one virus, the slide to the left shows the rapid evolutionary tree of this virus, often subtle mutations, but accumulating over time with already quite substantial diversity in that virus. So yes, it's one virus. Yes, it's got one common genome and structure, but there is profound variation in that. And indeed, one mutation, the GD substitution at 164, has given a subtle advantage to the G variant, which has allowed it to infect cells a little bit more readily. And that's already become the dominant form around the world. Next slide, please. So here we have it. We've got lots of genome sequences, and you can see over 1,500 nucleotide level variations, 740 different amino acid changes, and so forth. But fortunately, not much affecting differences in outcome. Next slide, please. So is it one illness? If it's a slightly different spread of viral genomes, does it still cause one illness? Well, firstly, infectivity changes. You all know about the R number, the famous R number, the number of people that can be infected on average by one person. And you'll see at the beginning of March, those R numbers in different regions in Britain were very high. And by mid-April, they were very much lower. Now, that raised the question, why? Is this due to changes in the virus and its mutation? And the answer isn't. This was, of course, due to lockdown. The capacity for any one person to infect more people was dramatically limited. If we hadn't known that, we would have had to have postulated that as one potential cause, along with postulating that maybe the virus itself had changed. But again, there is variation in region, and there's been variation over time. Next slide, please. Furthermore, of course, that infectivity potential varies in the social circumstance in which one finds oneself and what one's doing. So this is a classic case where one symptomatic individual attended a choir practice of 61 others. And because they were closely confined and because they were singing with forced voice, therefore aerosolizing and spreading droplet particles, that one person infected not 3, but 53 people, telling us a great deal more about how this virus was transmitted. So again, looking at variability, looking for the outliers, was able to tell us quite a lot in the early stages about how this virus might be spread. Next slide, please. But there's a lot more variation in this. We don't all get the same disease from contracting the same virus, even at the same load. And I've given you a reference there if you were particularly interested. Next slide, please. So the first thing is that broadly, I've said asymptomatic, but if we're looking at very mild symptoms, such that people might dismiss them, or no symptoms at all, somewhere between 47 and 70% of people will have very trivial symptoms or none at all, leaving only 30 with moderate to severe symptoms that are noticeable. Now, again, quite marked variability, but even amongst those that become symptomatic significantly, 24% of the 30 will manage themselves perfectly well at home, and a much smaller proportion will become sick enough to go to hospital. Next slide, please. We can then start exploring why that might be. And there are a number of factors. Here's one that flagged up very early. obesity is a strong risk factor for having severe enough illness to attend a hospital. So if we look at normal weight individuals, the proportion was very low. And if we look at those who were really substantially obese, the risk was very, very much higher. In fact, close to 4 times higher. Other factors have been identified. We'll come to those later. Next slide, please. Now, inside the hospital, there's variation again. A lot of people survive to go home. Some end up going to an intensive care unit. 17% of the 6% that come to hospital. In other words, only around 3% in the early days of those with symptoms. Quite marked variation. Next slide, please. What are the factors that put you on an intensive care unit? Well, actually, age is a factor for getting sick with significant symptoms and for hospitalization. But once you're in hospital, that non-severe versus severe group, age is a very, very profound factor. In fact, it's probably the dominant factor in contracting severe disease or ending up on an intensive care unit. Next slide, please. Sex as well is also very important. If we go click again, I think we should get the sex data up. It doesn't come up, but men essentially are twice as likely to end up on an intensive care unit as men are. Next slide, please. Now, furthermore, once you've hit that intensive care unit, or if you're on a ward, we're told the mortality worldwide is somewhere between 2 and 3%. That's highly geographically variable. It's probably closer to 1% in Great Britain. Next slide, please. But of course, that related to the symptomatic cases early on because we didn't know about the patients who weren't presenting. Even so, let's look at the ITU and hospital mortalities. Next slide, please. And you'll see that there's marked variation again. Not everyone who hits an intensive care unit died. In the early days, it was around 43%. And as we'll see, that changed with time. Some live and some die. Again, marked variability, whether you get symptoms, whether you're sick enough to go to hospital, if in hospital you're sick enough to go to ITU, and in either environment, whether you live or die. Next slide, please. Age in this circumstance for death is, again, a very profound influence. Next slide, please. So if you are under 16 years old, your risk is less than 1 in 13 million. In fact, there have been 7 deaths under 19 in Britain, and all of those people were substantially ill anyway. Next slide, please. However, next slide again. If you're in your 50s and 60s, the numbers go up to 5 in 1,000. Next slide, please. If you're in your mid-70s, it's already going up more steeply again. These are worldwide data. Next slide again, please. And if you're over 80, you're 70 times more likely to die than if you're under 40. Next slide, please. Indeed, if you're over 90 and you contract coronavirus and get symptoms, 7 out of 10 will die. So age strongly influences this variability in presentation, hospitalization, intensive care admission, and ultimately death. Next slide, please. And I've mentioned these data before. Men are at much greater risk. Next slide, please. There are other variable factors if we're trying to understand this, and these are the only ones we've identified thus far. High blood pressure, that also associates, as does diabetes though, with obesity and so forth as well. And diabetes is a very substantial risk factor. It's mentioned on 21% of COVID death certificates. Next slide, please. But there's more to it than that. We can look at deprivation. And in fact, 75% of cases going to an intensive care unit are from social deprivation classes 4 and 5, the most deprived of individuals. And we know that ethnicity is also important, particularly at Southeast Asians, actually, that are at profound risk. But again, if we start looking at that variability, to quote Lindsay Edwards' data, we have to start understanding why. Is there an obesity issue? Yes, Southeast Asians, males particularly, tend to have central obesity. Could it be due to poverty that also links to ethnicity and so forth? Next slide, please. Geography matters. Again, poorer populations are more densely packed, sharing more houses. They tend to live in larger cities, greater family connectivity, greater risks, I've said, for diabetes and so forth. These factors probably interact in complex ways that would require Lindsay to explain for us. Next slide, please. But timing also matters. So here was the ITU mortality in Great Britain at the end of March. and it had fallen to 41.6% by the end of May. Next slide, please. Now, this is looking at the number of hospital admissions, and you'll see that the mortality had fallen over time. Next slide, please. The same applies to intensive care unit admissions. Let's look at the rate of intensive care admissions and mortality. Again, we need to explore these data. Was it that we were just completely overwhelmed with patients at the beginning? And that was certainly true. We just couldn't do our jobs as well as we might. Or was it also the fact we'd learned something later on, which is why the mortality fell? It's likely to be one of those two things. Mortality now is lower again. Next slide, please. So the real value in trying to understand this disease and how to treat it and how to set population policy is looking at the differences between individuals, not in the similarities. Next slide, please. We can look, therefore, at which intensive care unit. We're trying to dig this out now. Some units seem to do better than others, and we need to try to understand why that might be. If we could work that out, we could learn from those units, and Dan and I could all do a better job than we were perhaps doing before. Next slide, please. We can look, this is the marked between centre variation, which is really demonstrated on that first left panel. Next slide, please. And again, please. If we also study the variation, it can help us understand the mechanism of disease. So we know now that ACE2 is the protein to which the spike protein binds from the virus itself. It's how it gets into cells. We can then try to say, well, why is it that children are at lower risk? And one factor comes up very clearly here. Children express a lot less ACE2. Next slide, please. We can start saying, why is it that women are at lower risk, and start looking at their immune responses. We find they get a much lower pro-inflammatory response, but a much better T-cell response. And these are the cells that clear virally infected cells from our bodies. So women have an innate biological advantage. And people like Lindsay and drug companies can start exploring these as potential mechanisms to harness therapeutically. Next slide, please. We can start doing blood analyses, as Lindsay sort of explained, to look at different types of immunity. And we'll move this quickly through to the next slide, please. And if we start looking in the way that Lindsay's described at expression of diverse proteins in cells, we can see that they start clustering. You can start identifying a specific signature for infection, but also starts to find clusters that define severe disease. Next slide, please. And indeed, if we look at those clusters even more, we can start seeing which ones are most associated with severe disease and death. And again, by looking at the mechanisms, this high pro-inflammatory cytokine response, we could start looking for targets that might improve outcome in us all. Next slide, please. And furthermore, we can see that this matters. If you mount a profound immune response and get very sick, but you don't get the T-cell response to clear the cells, you get severe disease. And in fact, viral load continues to rise when you're in a hospital bed. Whereas if you have moderate disease, the inflammatory cytokine response is less, you have more T-cells to clear the virus, and you do successfully clear the virus much more quickly. Next slide, please. We have to start then unpicking things. Is immune function driving a meaning that you get more likely to be infected if you have poor immune function, or does it drive disease severity in time? And we haven't got time to discuss this, but it does warrant a consideration about which came first in driving the disease or killing you with your response. Next slide, please. And finally then, failing to look at diversity and just looking at populations overall is a disaster. If you just treat things as being one disease, you'll get in trouble. We locked down late when we'd already let this virus get embedded in the community. Next slide, please. And again, because we didn't look at variability, we've had a one-size-fits-all policy, and everyone has locked down. We shut down schools, when actually early on, of course, we hadn't learned, but we do now know that children themselves are not terribly much at risk if they contract this virus. Next slide, please. We didn't shield specific populations. We exported the virus back into old people's homes. Obviously now, knowing what we know, we might actually allow children to run wild and get infected whilst trying to shield those who are at greater risk, the older, the obese, or the risk populations. Next slide, please. We need to understand the reasons for risk so that we can moderate it. Next slide, please. And we haven't yet done this. We still don't know the interaction of genes, environment, and comorbidities and how those interact cause variability. Next slide. We still don't know the reasons for variable outcomes between hospitals. We genuinely don't. And we're trying to do that work right now to see if we can learn why one hospital did better than another. Next slide, please. And we still don't know, next slide, please, the disease mechanism in terms of cause and effect of disease, as I've just outlined. Next slide, please. That is still an unknown. So to finish then, the value of studying disease populations isn't in looking at their similarities, but is in exploring the differences. And I think that's where I end. We have overrun. We have, I think I'm going to look and see whether we're allowed to get, to take a few more questions. Rosie, are you happy if we try to overrun a little bit to take some questions?
Hugh, that's fine. That's fine. Let's see if we've got some burning questions. The questions will all be answered. Some of them will be answered offline if we don't have time online.
Tremendous. Would you like me to take a couple now?
Yes, please.
Okay. So some questions then have come up on the panel, and there was one question asked about what is asbestosis. Well, it's a disease from inhaling asbestos, and it can cause scarring of the lungs and also mesothelioma, which is a malignancy. There was a question earlier on to Claire about what the normal population was to whom you were comparing your oil refinery workers. Would you like to be able to answer that one, Claire?
Yeah, of course I can. So the general population that we used was the population rates from England, Wales and Scotland. So what we used is national cancer incidence and mortality rates for these particular areas, and we stratified that by age. We just had males, and we did also stratified it by calendar period as well. So that was our reference group, was the population values from England, Wales and Scotland for mortality, particular mortality types, and for cancer incidences as well.
Thank you very much indeed, Claire. There's a question that's been asked to me about time series analysis. So how do you tease out the changes in a changing landscape such as a pandemic? And it's an excellent question, and it's one thing we have learned. We really do need constant data pipelines, because otherwise we're only, to take Lindsay's point, only storing the data that we think are going to be important, and we're ignoring the variability that we might only learn later to have been important. So I think we have to collect pretty much all data in an agnostic fashion, such that we can detect trends over time and in those variabilities. So I think it's a question of not being parsimonious in the data that are required and doing time series analysis on them. And no country has proper infrastructure to do that. And if there's a learning for the health service in Britain and elsewhere in the world, it would be to create such pipelines. Dan, I see a question for you about human space exploration and hypobaric and therefore hypoxic as well, long-term missions. The question is there any experimental data or other experimental data on short or long-term human adaptation to hypoxic and hypobaric environments, or to hypobaric environments with elevated oxygen concentration, which of course is interesting, isn't it? Drop the pressure, increase the FiO2.
So, of course, and you know, the examples I've used there with the populations who've evolved at high altitude are clearly evolved to living in a low pressure environment. However, it's combined with low oxygen. But we have a lot of adaptive responses, as I've explained, to hypoxia. What we know much less about is whether or not humans have the ability to adapt to high concentrations of oxygen and the oxidative stress that will result from that. We have only a very limited ability to, with our innate antioxidant systems to combat that. I'm not aware of data looking at long-term adaptation to high concentrations of oxygen, but there is certainly a potential for harm there.
And thank you. Just to pick up on Dan's work to show that the study of variability is important. It was a 20-year journey for the team. But what it has ultimately shown is that hypoxic adaptation in humans is metabolic. It showed that muscle wasting and protein synthesis suppression that Dan referred to in crucian and carp actually does happen in humans and is utilized in critical illness. And it's also dependent on changed substrate utilization. And that is modifying the way we're going to be treating our patients in intensive care units. So studying this sort of variability does have real direct translational value to human health and disease. I think we've picked up most of the major questions there. There's lots of opportunities to post more, and we may be able to pick those up in the discussion section later on. But so that you've all got the chance to get a quick cup of coffee before we move on to the rest of the morning, I'd like to conclude by firstly thanking Mike Tipton, Igor Mekjavic, Rosie and the others at the Physiological Society for putting the symposium on, for the excellent talks we had this morning, which covered great diversity from machine learning, occupational health, evolutionary adaptation and more. And then to thank you finally as well for all of you for attending. Let me remind you to use the social media, the hashtag Variability2020, to post other questions that might be picked up later, to join the networking sessions later and the statistical discussion section later, if that's what makes you interested. Mike, any last words for you or from you?
No, other than to thank you very much, Hugh, for what was a first-class session. And people should know that, as you say, they can continue to ask questions. The questions will be addressed offline. The whole of the conference is being recorded and will shortly be available on demand online, as will the book of abstracts. So there's lots of opportunity to follow up on what necessarily had to be a condensed session. We could have done a whole day on that, I have no doubt. Thanks again. So we move now on into a different environment, but still one where variability, its presence and its absence are critical, as we shall see. So the vast majority of people who engage in physical activity on the planet still do so going to work. In fact, the area of sports science that many of you engage in began as work physiology. Occupational physiology is an underrated but very important area of what we do. And so we'll start by looking at occupational related problems, and then we'll move on to looking at even in an engineered system where there's a meticulous attempt to reduce variation, it can still occur, and we'll learn what needs to be done to reduce it. So our first speaker is Urša Ciuha, and along with Professor Igor Mekjavic from the Jozef Stefan Insitut in Slovenia, and the other co-author on this paper is Andreas Flores from the University of Thessaly in Greece. So over to you, Urša.
Thank you for the introduction. So if we can just move to the next slide, please. So just briefly, the perception of thermal comfort is primarily established by the cortical integration of information from the skin and from deep body regions. So this information actually initiates behavioral actions before activation of more metabolically demanding responses, such as shivering and sweating. But in some working scenarios, and especially in industrial settings, these behavioral modifications might not be possible, which can lead or compromise thermal comfort. So this can have a detrimental effect on working productivity, but can also affect health and well-being of people working in such scenarios. So it is well known that perception of thermal comfort is actually highly individualized, which means that people do not perceive the same temperature equally. What can affect our thermal comfort is age, sex, our previous experience, also season, the level of physical activity we're involved in, and so on. So the next slide, please. So the main question of the present talk was what actually drives the thermal comfort. So to answer this question, we gathered the data from several laboratory and field studies, which included information on physiological data, environmental data, anthropometric data, but mostly and most importantly, subjective data. So the participants in these studies actually provided information about their thermal comfort and thermal sensation. So the next slide, please. So once all of this data was gathered and put in a right format, it was further analyzed. So we had to decide whether we would go with a standard statistical approach, which would actually look at statistical significance of the measurements at different levels of thermal comfort. So actually the measurements that would vary significantly between different levels of thermal comfort would present the most important factors of thermal comfort. But what we decided was to use the machine learning approach. So with this approach, we actually treat the levels of thermal comfort as classes that need to be predicted. This means that the measurements actually present classification features. So in this manner, we can actually present the results in a form of decision tree, which was also done in the case of the present talk. So the next slide, please. So what we can see here is a decision tree. This one is based on the data that was taken from the laboratory studies. So we can see the nodes, and each of them actually presents three things, as you can see. So the upper one presents or shows the predicted class. So it means that whether we are talking about comfort or discomfort. And then the next one presents probability of discomfort, which is indicated by a number 0. Let's say 54 on the top, which you can see. And lastly, the node also presents the percentage of observations in each node. So on the right side, you can see the input parameters or classification features that were actually included in the analysis. And the most important ones are the ones in bold, and these are also found in the decision tree. So if we now go to the tree and we look at on the top, we can see the RH micro, which means microclimate relative humidity. So if the microclimate relative humidity is below 60%, we follow the left line and we end up at the blue node, which says comfort of 0.20, which means that there is a 20% chance of thermal discomfort in this case. But if we follow the right line, which means that the microclimate is above 60%, then we can see that there is a 63% chance of thermal discomfort, and this is according to 78% of all participants. If apart from this microclimate relative humidity, the chest microclimate relative humidity is above 80, then we again follow the right line and we end up at the node which says that there is a 77% chance of thermal discomfort. And this is according to 37% of all participants. And if apart from those two factors, also the heart rate is above 74, which is RH and indicated by RH, then we again follow the right line, and we can see at the end on the right corner that there is a 80% chance of thermal discomfort, which is according to 34% of all participants. So the factor on the top, microclimate relative humidity, is the most important factor, followed by the rest of the factors, which actually build up and at the end present increase in thermal discomfort. So the next slide, please. So the next decision tree was actually based on the field study, from the data from the field study. And again, we can see nodes that present the same thing as in the previous slide. And we can see the input parameters or classification features on the right, which were the measurements that were actually considered in the analysis. The most important ones are again in bold, and these are also located in the tree. So if we go to a tree now, we can see on top, TR is actually air temperature. And if the air temperature is below 29 degrees Celsius, we follow the left line, and we come to the note which says comfort of 0.11. So this means that there is only 11% chance of thermal discomfort. But if the air temperature is above 29 degrees Celsius, then we follow the right line, and we end up at the note which says discomfort of 0.61, which means that there is 61% chance of thermal discomfort, which is according to 68% of all participants. If apart from the ambient temperature, the RP, which is perceived exertion, is above 9, then there is a 79% chance of thermal discomfort, which is according to 42% of all participants. And if apart from those two factors, BMI, which is body mass index, is above 26, then the chance of discomfort actually raises to 95% chance. So the next slide, please. So to summarize, based on the analysis, which was machine learning approach, we can say that thermal discomfort is primarily affected by exogenous factors, which means that it is affected primarily by the surrounding environment, which is ambient and microclimate. And secondly, by endogenous non-thermal factors, which include body mass index, heart rate, and other factors. So with this, I would conclude, and thank you for your attention.
Thank you very much, Urša, for a very clear and interesting presentation. We'll move straight on now to Chris Gray, who works for Infinium, but is representing the Energy Institute here. Chris has been a statistician for over 30 years. I suspect longer, because I think statisticians are born, not raised. So Chris is going to tell us about the importance of variability within the engineering systems. Thank you.
Thank you very much, Mike, and thank you for the invitation to this conference. It's been fascinating to hear about all the different takes on variability in different situations. So I'm going to take a look at how, can we have the next slide, please, Rosie? I'm going to take a look at variation in the measurements used in the fuel industry to monitor the quality of our fuels. And in part of that, we'll be looking at precision statements, which describe variability and what they look like, and looking at some challenges that we have in making them fit reality. Next slide, please. So if you go down to the fuel pumps, you'll see that above the pumps, there will be the standards which the fuels comply to. In Europe, you'll see that they're EN 528 for gasoline or EN 590 for diesel, and these place various specifications on the fuels. A couple of the tests or characteristics for diesel fuel that are important are the cold filter plugging point, which protects your vehicle at cold temperatures. At cold temperatures, crystals can come out of the diesel and block your fuel filter, and after a while your car will come to a halt. You can see a picture of some of those crystals on the right. Needles that you see there are good things and typically come after the fuel has been additized to affect the crystallization. But if you don't put additive in there, they can come out as plates which block a filter very quickly. So that's one test, and it's measured as a temperature at which that filter gets plugged. A second test we'll look at, the high friction reciprocating rig, HFRR. This test rubs a metal ball against a plate for 75 minutes until a wear scar is created. And you can see pictures of those scars there on the right. If you don't put additive in the fuel, then the fuel won't lubricate the diesel fuel pump adequately, and you may wear it out really rather quickly, which is a very expensive replacement. So my interest in Vinium is that we're a supplier to both the markets for these additives. Next slide, please. So how do precision statements and stuff get formed? What's the process? Well, industry committees develop procedures. Once they've got them in place, there is a standard called ISO 4259, which tells you exactly how to run and analyze an interlaboratory study to generate a precision statement. So that will include a lot of stuff about what outlier test to perform, how many laboratories and samples you need, and all sorts of things, laying it down very precisely to create a good outcome. So typically you might have 6 to 10 laboratories with 8 to 10 samples, each laboratory testing each sample twice. Once you've got a suitable precision statement that describes the variability, you want to make sure that all the laboratories comply with it. So you, in preparation, there's part 4 of ISO 4259 that describes statistical quality control methods that individual laboratories can use by running the same sample, making sure they get the same result. And there are the Energy Institute and other organizations run proficiency testing programs. Laboratories monitor themselves by getting blind reference samples. Every laboratory tests it and you compare results. And also part 3 of ISO 4259 describes how you can take all that data from the PTPs and check that it's consistent with the precision statement. Finally, the last box, you have a part 2 of 4259 describes how suppliers and vendors can use it to check them producing the right quality of material. And if there's a dispute, well, they can use the watertight, legally binding resolution process that's in there to decide who is in the right. Next slide, please. So what does a precision statement look like? It will define 2 things: repeatability and reproducibility, which describe the level of variability. So repeatability is the difference you might see between 2 samples tested under the best possible conditions. You've got, in a short period of time, you're using the same operator and equipment. You only expect to exceed that 1 in 20 times. Reproducibility is similar, but you're testing at different laboratories. ISO 4259 always assumes that the data have a Gaussian or normal distribution, which means that these R and R are 2.77 times the corresponding standard deviation. So you can see below some precision statements for these tests. For HFRR, the repeatability is constant. It's either 50 or 70 across the range of the HFRR, depending on whether the measurement's made by camera or visual. So the camera is more accurate. On the other hand, CFPP, as you go down in temperature, repeatability and reproducibility increase linearly. And power law forms are also permitted as well by the procedures. Next slide, please. So how well does this structure fit the actual data that we get when you go back and look at it? So this is for HFRR, you'll recall that the precision was meant to be, the precision statement says the precision is constant no matter what the level of wear scar that is measured. But if you look at the graph on the left, you'll see that reality is far more complex than this. The precision increases up to a point, about 450 or 500, and then the variability remarkably improves suddenly and continues at a low level. The precision statement sticks a flat, straight line through all of it. So reality is a a lot more complex. Why is that happening? Well, the causes are being investigated, but even if we find the causes for why that is happening, it's not clear that we'll be able to alter the precision statement easily and and still have an effective dispute resolution process. Next slide, please. So the CFPP test, how well does the precision statement work there? Well, CFPP, as the temperature went down, the variability went up according to the precision statement. But when you look at the graph, you can see that whereas that is true in general, there are quite a few outliers where the variability is much higher than would be expected. These the distribution here also can be non-Gaussian. It can be quite bimodal in character, which is very unusual. And this is due in some way to the crystallization process, which is very non-linear. And the more that the crystallization process is adapted through the additives which are in there, the more the risk of higher variability. The precision statement works on average, but it doesn't, for other fuels, it doesn't work very well at all and can cause confusion. Next slide, please. So we have some challenges. We have a mature product quality system in the fuels industry. It minimizes the risks to motor vehicles, and the whole process works well. But it is, there are situations where we haven't captured the variability as well as we would like. And for particular fuels, there can be large deviations in reality to what the precision statement would predict. And it would be good to improve those without compromising the whole integrity of this dispute resolution process. Thank you.
Thank you very much, Chris, for a very clear presentation, and interesting to see how variability is even plaguing your part of the work arena. We have some questions coming in from actually some of the panelists. We'll start with you, Chris, if that's okay. Urša, can we see you as well? Because I think I've got one for you. So one question is, do the standards 4259, for example, define the threshold of acceptance or the maximum variability allowed? What's meant by the term exceeded that you used? That's for Chris.
They do not describe the maximum variability that is permitted. They will specify the minimum size of experiment that must be carried out in order to quantify the variability sufficiently precisely. There are other standards which other organisations look at to ensure that there is sufficient discrimination in a test method that it is useful. If it can't discriminate between poor products and well-performing products, then it's not useful to employ it.
And excuse my ignorance, but in the standards themselves, are the thresholds for pass-fail at a set point, or is there a sort of a fuzzy area?
There are no. There is a hard limit. So these, for example, for the HFRR test, there's a limit of 460 microns in Europe and the rest of the world, and 520 in America. The CFPP, well, that depends on how the temperature in the country. So for Britain in the winter, that would be -25 degrees C, but around the world, it can vary substantially.
Lovely, thank you. And one quick one for you, Urša. We do quite a lot of preliminary tests on people before we accept them to be participants into experiments. Those tests are nearly always medical and physiological. Should we be testing them psychologically as well before we admit them to experimental cohorts?
Yeah, that's actually a good idea, because sometimes, yeah, when you start the experiment, you actually realize that some people perceive comfort in very odd situations, such as very high temperature or very low temperature, high humidity, and so on. So yeah, basically that would be a good idea. And we actually did that in one of the studies before, and yeah, it makes sense, because then you get people that actually report on what they actually it's better to ask people about their perceptions before, so you know how to proceed in the study then later.
So the next thing we need to do is find out what standard test we can use to include and exclude people as a pre-screening test. So yeah, that's something for people to think about. We won't debate that now. Thank you to both our speakers in the industrial session. That's fantastic.
Thank you.
Thank you.
Thank you. So we now move on to what is the icing on, I think, a very rich cake. And I know lots of you over the 2 days have had questions relating to how you can deal with your own data. And this is the session where we will get some insight into that. And do remember that we hope to follow this up in about a year's time with the workshop where we can get deeply involved in individual data sets. But I want to introduce now the chairman of the statistics session, and that's Gordon Drummond, who is Honorary Clinical Senior Lecturer in the Department of Anaesthesia and Critical Care at the Royal Infirmary of Edinburgh. Over to you, Gordon.
Thank you very much, Mike. I have been most impressed by the variety of material that we've been listening to so far, and I hope we can keep that going in this session. Before we come to the discussions, I've got a double act to introduce of Alan Batterham and Greg Atkinson, both from the same department, talking about very slightly different projects, a little bit of variability, on response heterogeneity. And I think it would have been actually quite useful to have heard them talking yesterday, because a lot of what they're going to say, I'm sure, will be highly relevant to what we're going to be discussing later as well. So without any further ado, I'd like to introduce Alan Batterham first on quantifying treatment response heterogeneity in parallel group trials, which of course is the usual sort of study that is done in physiology and in medicine. Alan, please.
Thank you, Gordon, for the introduction. Double act about right. I'm the warm-up man in this case, I think. Okay. Thank you to the Society and the organisers for the invitation to present at this excellent workshop. I'll give a little background first, and then I'll move on to running through a basic analysis of data from a parallel arm randomized controlled trial. Sorry, Rosie, back a slide, please. Thank you. Stay on that one for a bit. I'll run through a basic analysis of a parallel arm randomized controlled trial, just to point out some of the key issues as we try to quantify heterogeneity of treatment effect in this setting. I think the presumed promise of personalized medicine has sparked a resurgence of interest in non-constant treatment effects or heterogeneous treatment effects. And there's been a proliferation of terms in the literature for this notion of better targeting treatments to particular individuals or groups based on their differential responses observed in randomized trials. These terms include personalized medicine, precision medicine, stratified medicine, or personalized public health, precision public health, stratified public health. In fact, just insert your subject area of choice. Next slide, please, Rosie. This proliferation of terms led Stephen Sen, who's a statistician who's written a lot in this area, to wonder what's next. And he proposed multi-omic medicine, super-focused medicine, hypergranular medicine, or I like this one, digisymbiotic medicine. And these terms will doubtless appear in the literature soon, if not already. Next slide, please, Luke. Another tweet by Stephen Sen illustrates the fact that many people believe that the promise of personalized or precision medicine has been overblown somewhat in the literature so far, and that it represents more hype than hope in many instances. And in some quarters, there's a belief that often the best indicator of whether a treatment will work for individuals is indeed the mean effect. That's not to deny the potential for personalized medicine where we are able to quantify heterogeneous treatment effects. Next slide, please. The statistical pitfalls of personalized medicine are laid out very clearly in this 2018 article by Stephen Sen. And if you don't follow Stephen Sen or read his work and you're interested in personalized medicine, variation and heterogeneous treatment effects, I urge you to read everything that Stephen Sen has published, and you'll certainly learn a lot from it as we have done. Sen identifies the pitfalls of personalized medicine, and perhaps the major one is the arbitrary dichotomy of continuous variables, which Sen calls dichotomania because of its prolific nature in the literature. And I've just pulled out one quote from this paper, which is of interest to this particular talk. And Sen says the problem is that the distinction between these responders and non-responders can be arbitrary and illusory. Much effort then goes into the effort to uncover a trait to explain this differential response without assessing whether or not such a differential exists. And I think this is one of the problems in our areas and in sport and exercise sciences as well as the biomedical literature, is that there's often the assumption or presumption that the heterogeneous treatment effects exist. And then there's perhaps an awful lot of research waste as people go off trying to find out what the mechanisms are or the effect modifiers, etc., for these particular individual responses. We've always argued that the very first stage should be to properly quantify heterogeneous treatment effects and work out whether such a differential exists, and only then move forward to trying to explain it. We see this quite often in the literature as well, where people try to identify responders and non-responders by looking solely at the intervention group. So if they have a control group, they use the control group to assess the mean effect as intervention minus control. But then some researchers have forgotten about the control group when it comes to identifying variability in response and have focused solely on the intervention group, just looking at whether people have responded or not responded beyond some threshold. And we would argue that that is a flawed approach to identifying heterogeneous treatment effects. Next slide, please. In our reviews that we've conducted so far, and these two published in Experimental Physiology in 2015 and 2019, we've focused on parallel arm randomized controlled trials and focused on identifying heterogeneous treatment effects by comparing the response variances in the intervention group and the control group, which I'll say more about in due course. Next slide, please. We've applied these approaches in a couple of areas, and the first one was a critical review on maximal oxygen uptake, and the second one is a systematic review and a meta-analysis of randomized controlled trials looking at inter-individual differences in weight change following exercise interventions. Both of these reviews were led by a former PhD student, Phil Williamson. And you can just focus on the second one just for a minute. In common with other recent meta-analyses in different areas, we found little robust evidence of heterogeneous treatment effects for the variation in weight change following exercise intervention. So in that field, the assumption is often made that weight change is heterogeneous between subjects, And there's a lot of effort being spent on looking for mechanisms of that heterogeneity to try to explain it. But we and others have found no evidence or no robust evidence of heterogeneity of weight change, at least following exercise intervention. Next slide, please. Our thoughts have been in this area have been influenced by a number of papers. I referenced all of the work from Stephen Sen, but I also would like to highlight these two papers. At the bottom, individual response to exercise training, a statistical perspective, and Anne Heksteden presented yesterday an excellent talk giving an overview of her thoughts on this area. And the one at the top is an invited editorial on this particular paper. So I'd urge you, if you're interested in this area, to read both of these, and they'll be very insightful. Next slide, please. So this is a little bit of a busy slide. I'll walk you through it. The table at the top is from Katie Bell and colleagues, and at the bottom is from Stephen Sen's Mastering Variation paper published in Statistics in Medicine. Assessing heterogeneity of treatment effect in a randomized trial is all about partitioning variance. That's what we're talking about throughout. And if I draw your attention to the table at the top to begin with, the table 2, sources of variation in measured outcomes in randomized controlled trials. In a parallel arm randomized controlled trial, the typical focus is on T, the between treatments variation, in the comparison of the mean effect in the intervention group minus the mean effect in the control group, so the average effect of the treatment. And most papers don't go beyond that mean effect. But variation around the mean effect in the control group, the response variation, is given by B, the between patients variation, and W, the within patients variation. Whereas variation in the response around the mean in the intervention group or the treatment group is given by B and W, the same as the control group, but also by R, the between patients variation in treatment effect, or variation caused by differences between patients in the effective treatment. So arguably, in a parallel arm randomized trial, if we subtract the response variance in the control group from the response variance in the intervention group, we will get R, the between patients variation in treatment effect. Now, if you just scoot down to table 1 from Sen's Mastering Variation paper, the table at the bottom, one of the issues that was covered by Anne Heksteden yesterday is that this variation, R, in the top table, also includes the variation from occasion to occasion when the same patient is given the same treatment. Now, we can't access that in a randomized controlled trial because you're only given the treatment once and measured before the treatment and measured after treatment, usually for a continuous outcome. So we can't fully isolate the patient by treatment interaction, which is the C in Sen's table, the extent to which the effects of treatment vary from patient to patient. To fully isolate the patient by treatment interaction, you would need a replicated crossover design, which is the subject of the next talk. However, for long-term, relatively permanent chronic adaptations, we argue that the replicated crossover is not appropriate because you could never have, arguably, an adequate washout between the repeat applications of the treatment. There will always be some residual effect, if not detectable in the outcome variable, then at least at some deeper cellular or molecular level. So for longer term chronic adaptations, the parallel arm randomized controlled trial is likely the most practical, pragmatic approach to trying to identify mean effects and also the heterogeneity of treatment effects. And we agree with a statement in a recent paper by Cortez and Stephen Sen and others that the burden of proof should be on providing some robust evidence for heterogeneity of treatment effect before we advocate for precision medicine. Next slide, please. Okay, so having done some background on parallel arm randomized controlled trials and the notion of subtracting the response variance in the control group from the response variance in the intervention group to try to pin down heterogeneity of treatment effects, I'll just walk through an analysis of some data from a randomized controlled trial. In this data excerpt, we've got a randomized controlled trial, independent groups, parallel arms, and we've got 100 participants per group. In the data set you've got there, you've got ID on the left, then you've got group coded 0 and 1, and this is 0 for the control group and 1 for the intervention or the treatment group. The outcome measure is systolic blood pressure at 6 months, and that is measured at baseline, which is your pre-measurement in this table, and measured after the intervention, which is your post-measure. I've also put in the change score, and you'll see on the far right of the table a dummy variable called X bar, which stands for extra variance. That is coded in the same way as group, and it's the variable that we're going to use in the analysis model to allow for different response variances in the control and intervention group, and to quantify the difference between the two with a mean difference in variance together with its confidence interval. And that will be the true individual response variance, where true implies free from measurement error, random measurement error over the 6 months of the study. Next slide, please. So what I've got here is some stripped back SAS code, SAS software, just to show the analysis. Now, it doesn't matter that it's SAS, you can reproduce the same analysis in Stata or R or SPSS or any of the other packages that allow linear mixed modeling. So we run these analyses typically using linear mixed models, which is prop mixed in SAS. We're using restricted maximum likelihood to get unbiased estimates of the various components, variance components. The first thing I'd like to draw your attention to, sorry, I was getting some feedback there. The first thing I'd like to draw your attention to in the red box is the no bound command. And the no bound command is an important one because usually variance components are constrained to be positive based on the premise that you can't have a negative variance. But in this case, we release that constraint and allow the variance to become negative, either in the mean or in the confidence limits. And that better reflects the uncertainty in the estimates of the response variances. And also in this context, it has a specific meaning in that a negative variance would imply a greater variability in response in the control group versus the intervention arm. Whereas I've said it's a greater variance in the response variance in the intervention arm versus the control that implies heterogeneity of treatment effect based on that partitioning of variance that I went through earlier. We'll move on to the model. The model has the post-intervention, the after the intervention systolic blood pressure as the dependent variable. It has group as the fixed factor, so the difference between groups in the post-intervention blood pressure. And we also condition on the baseline or the pre-value, as this has been shown to be the most statistically efficient analysis by Frisson and Pocock, Stephen Sten, Vickers and Altman, and many others, that if you've got a reasonably reliable measure for your continuous outcome, then an analysis that conditions on the baseline is more statistically efficient. And of course, in expectation in a randomized controlled trial, the groups are expected to be equivalent at baseline in any case. An interesting thing here is that if you've got baseline in your model, so the pre-score as specified here, it doesn't matter whether you use the post-intervention systolic blood pressure as your dependent variable, or you use the change from baseline to post. They are mathematically equivalent, as demonstrated by Nan Laird many years ago. You'll get identical estimates if you include the pre-score in the model. But I prefer using the post-score as the dependent variable because the intervention occurs after the measurement of baseline. And so, of course, the intervention cannot affect the baseline score. It can affect the post-intervention score. So although it's equivalent, we prefer to put the post-score usually as the dependent variable. Then go down to the random statement. That's the fixed effects. And this is a mixed model, so it has fixed and random effects. And the random effect is where we put in our X-bar variable, and that is the allowance for different response variances in intervention and control groups. And this is the variable that allows us to quantify the heterogeneity of treatment effect as the difference in response variances between the intervention group and the control group. And we're requesting all of that with 95% confidence interval. Okay, next slide, please. Okay, so here's some basic estimates. And first of all, this is the fixed effects. I will say that the baseline value, the mean baseline blood pressure in this trial was 133 millimeters of mercury. And you can see the estimates at 6 months in the control group are 127. And in the intervention group are 123.2. So the mean effect of the intervention on systolic blood pressure is the difference between those, which is 3.8 millimeters of mercury, so a reduction of 3.8 millimeters of mercury in the treatment group versus the control group. And you can see the p-value there is 0.0015, and the confidence limits are 1.5 to 6.2. The confidence interval gives a plausible range of effect sizes that are compatible with the data and model. So that range is a reduction of 1.5 millimeters of mercury up to a reduction of 6.2 millimeters of mercury, with the average being a reduction of 3.8 millimeters. So that is the average treatment effects. Next slide, please. Here are the variance parameter estimates, and this is the X-bar estimate. And the difference in response variances between intervention and control is 7.83. And there's the confidence interval for that 7.83. And this is where the no bound command allowed the variance to go negative. So we have a lower confidence limit of minus 21 and an upper confidence limit of 36. So you can see that there's a very, very wide confidence interval, and there's lots of imprecision in estimating the response heterogeneity. And this really reinforces the fact that you need very large sample sizes to pin down heterogeneity of treatment effect for continuous variables. And if you power a study on the mean effect with the usual 80 or 90% power, this is the sample size I need to detect a certain minimum clinically important difference, then you'll need at least 4 times that sample size in order to pin down individual response heterogeneity. That's assuming that your response heterogeneity is of a similar magnitude to the minimum clinically important difference. If you want to detect smaller amounts of response variability, you'd need many more multiples of the original sample size. So these trials don't often get done that have sufficient power and precision to pin down heterogeneity of treatment effect. And this has led some statisticians to argue that most studies will be better contributing to meta-analyses of heterogeneity of treatment effect rather than trying to identify it from a single study in its own right. And we and others have engaged in that process. Probably the best way forward in this regard would be IPD meta-analyses, or meta-analyses that use the individual patient data rather than meta-analyses that rely on summary statistics, which are the most common. But for the most part, trials would be unaffordable, I think, to try and pin down heterogeneity of treatment effect based on the requirements for precision. Next slide, please. So if you, in the code, if you replace the random XVAR with repeated group equals group, this allows you to pull out the separate response variances in the control group and the intervention group. And you can see here that the response variance in the control group is 66.9, 74.75 in the other group, and you'll have seen that that's just the difference that we've already identified. The difference between those is the 7.83. Now remember that that's a response variance, so to express this as a standard deviation, we have to take the square root. So the standard deviation for individual responses, as we call it, is 2.8 millimeters of mercury. Next slide, please. You can use the net mean response, which was a reduction in blood pressure of 3.8 millimeters, and the standard deviation for individual responses of 2.8 millimeters of mercury, to form a distribution of the true net treatment effect, which is what I've done here. So this is just a normal distribution centered on the mean effect of a reduction in blood pressure of 3.8 millimeters of mercury with an SD for individual responses of 2.8. I've put in the black line the minimum clinically important difference, which I've defined as a reduction in blood pressure of 3 millimeters of mercury. This isn't plucked out of thin air. This is the value that the FDA often recommend in blood pressure trials. And it's also a reasonable value if you look at the epidemiological literature, looking at the association between blood pressure and morbidity and mortality. So I'll just for now put this in as an MCID of 3 millimeters of mercury. And we can then estimate the proportion of intervention participants, the proportion of people who would undertake this intervention, who would benefit versus what would have happened to them if they were in the control group, which is the counterfactual. And that is simply the area that's shaded in red, the area under the curve beyond the minimum clinically important difference. Next slide, please. So with the example data, the mean intervention effect is a reduction in systolic blood pressure of 3.8 millimeters of mercury. The standard deviation for individual responses is 2.8 millimeters of mercury, but with big confidence limits, of course. An individual drawn at random from the intervention group will have a reduction in blood pressure versus control of 3.8 plus or minus 2.8 millimeters of mercury. So the individual response SD is the typical variation around the mean response at the level of the individual. And then from that response distribution that I showed you in the last slide, the proportion of people undertaking this intervention benefiting versus control against an MCID of a reduction of 3 millimeters of mercury is 62%. But the confidence interval derived from analytic formulae at least is huge, ranges from 24 to 99%. So from around 1 in 4 people benefiting up to almost everybody benefiting. Those confidence limits, next slide please, are taken from Maya Makers and Tyler Wanderveel's paper, which was published in 2018 in Statistics in Medicine, applied in this case to random effects meta-analyses, but the principles are the same. The contributing factors to the confidence interval are the standard error of the mean effect, which is the squared, which is the variance in mu, and tau squared, which is the individual response variance in this case, and then Q, which is the minimum clinically important difference. It's much better actually to use bias corrected and accelerated bootstrapping for deriving the confidence intervals for these proportions. that even in modern times, this is a very computer intensive and non-trivial exercise. But that is the definitive method. Next slide, please. So my take-home message of this brief run through parallel arm trials is that parallel arm trials are likely the most pragmatic approach for examining heterogeneity of treatment effect for chronic, relatively long-term, relatively permanent adaptations. Oh, thank you. A control group counterfactual we regard as a sine qua non for isolating heterogeneity of treatment effect. We recommend analyzing these data, like the ones I showed you, using linear mixed models conditioned on the baseline. You can derive the true treatment response distribution in the way I've described, and derive the proportion of responders with a confidence interval. And I urge everybody to embrace the uncertainty in the estimates, and just emphasize again that a very large sample size is required for adequate precision for estimating heterogeneity of treatment effect. I am Michael. Thanks very much.
Thank you very much, Alan. That's a very comprehensive review of, as we both agree, the commonest sort of study that we come across. We're going to move on now to the replicated crossover trial, which may in fact answer one of the questions that's been posted on the Q&A board, quantifying treatment response in replicated crossover trials from Greg Atkinson. Greg, please.
Thanks very much, Gordon. And thanks to Alan, who's already presented on quantifying treatment response variability in parallel trials. And these are the most appropriate design for chronic or longer term responses. So I'm now going to talk about the situation of acute or shorter term response variability, which I hope to show can be explored using a replicated crossover approach. So let's start by looking at some typical data. If Rosie just advances to the next slide. So this is data taken from a conventional 2-trial crossover study. And here we have acylated ghrelin, which is measured pre and post in 2 experimental conditions. These being a 30-minute period of exercise in one condition and a 30-minute period of rest, which is the control condition in this case. So exercise tends to decrease levels of ghrelin, but you can see that we seem to have quite large individual differences in response to the exercise. And maybe I can even identify so-called high responders or non-responders on the basis of where their responses lie in comparison to a threshold for clinical importance, or the MCID, which is labelled on that graph. I've just set this rather arbitrarily at a reduction of 15 picograms per milliliter. So this all looks really encouraging for individual response variability. I'm sure you've seen this sort of plot in the literature, but things might change once you're able to look at what lies beneath these observed data. So next slide, please. So I'm very good at this. I can tell you that I actually made the data up, and I purposely engineered there to be no individual differences in response whatsoever in the data set that you saw previously. So we have true responses, which I've engineered to be a drop of 30 units for everyone. And then we have normally distributed test-retest errors that are added to those true values to give the observed data. This is what could be underneath any observed data, I suppose, that is collected in a conventional crossover study. Unfortunately, there's little you can do about this uncertainty in what lies beneath the observed data, at least from a data analysis perspective. And to appreciate this, I think like Alan did, we have to look into all the components of variability that are at play in that data set. So next slide, please. So Alan has already shown these components of variability in his presentation. This table was put together by Stephen Sen to show all the components of variability that can hide underneath the observed data. And I draw your attention especially to the inability to partition out true response variability, which is labeled component C here, from within person test retest error, which is labeled component D in Stevenson's table. And that's all if you have a conventional crossover study. And this is because any subject by treatment interaction component, which is the nitty-gritty of response variability, that can't be separated from the test retest error component. Next slide, please. So let's look at the data in a bit more detail and explore it in a bit deeper fashion. We can look at the data that I simulated and I've already shown you in the previous slide. Next slide. So let's start in the conventional way by quantifying the mean treatment effect, which Stevenson labeled component A in his table. So here we might use a paired t-test to quantify the difference in mean change between treatment condition and control condition. For convenience, let's ignore any exploration of carryover or sequence effects in the trial when we're doing this. So importantly, from the paired t-test results, we get a mean treatment effect, which is essentially the difference between the two conditions in the changes, in the pre-post changes. And we can see that this is around about 30 picograms per milliliter, which is exactly kind of what I simulated in that data set. We've also got the associated confidence interval and a p-value. Next slide, please. I'll just pause for a bit while we're considering this mean treatment effect. I just want to highlight that counting how many responses are above or below a threshold, and this happens to be very popular, this might tell us nothing about individual response variability. And this isn't like the approach that Alan just showed in his presentation. This is actually counting the observed number of people who fall above or below a threshold in the samples of interest or in the conditions of interest. Pretty ironically, this particular responder counting approach might merely be another way of showing differences in the mean treatment effect, and it might tell us nothing about response variability at all. So here we see that if the mean change differs between groups or conditions, so of course does the area under the curve of observed changes relative to any response threshold. This is nothing to do with treatment response heterogeneity as defined by whether individuals differ in their response to the same intervention, which is usually of greater interest. Next slide, please. You can pretty much see here when I mentioned this particular issue and how common it seems to be becoming, especially in the exercise science literature, you can see what Stephen Sen thought of that. Next slide, please. Anyway, back to our data, and let's delve a bit deeper into the analysis of that data. The paired t-test or repeated measures general linear model is synonymous with a two-factor general linear model, whereby one factor is subjects entered as a random effect. This is what the paired t-test is doing. It's just partitioning some known variance due to subjects or patients, because each subject's change is measured in the two conditions. So we get the same mean treatment effect as we did before with the paired t-test, but in this model we can also get a variance component for between subjects, and that's like component B in Stevenson's table. But just to highlight that this is still not any indication of individual response variability. Next slide, please. So what this is mathematically is simply the average of the response for each person in the two conditions. So it's merely the variability between people in the mean change that's been averaged across the intervention condition and the control condition. So it's still not getting to that subject by treatment interaction to the response variability itself. Next slide, please. So let's delve a bit deeper and try and get to this really important subject by treatment interaction term, which would really tell us if there is treatment response heterogeneity between subjects. This is the important component C in Steven-Sen's table, which we are trying to separate from the component D, which is the test-retest error or the measurement error. Unfortunately, this question is impossible to answer with a conventional crossover design. The subject by condition interaction term cannot be obtained because there's been no replication of each of the experimental conditions. Therefore, it's really impossible to partition any response variability away from the within subjects error component. So you can't really separate the components C and D that Steven-Sen labeled. Next slide, please. What may be interesting is the variance comparison approach that Alan has covered in his previous presentation. Here we see that the standard deviation of change for the two conditions, these are pretty similar, in agreement with the fact that I engineered no response variability at all in this data set. And just to say, you can see that there's a minus or a divide sign in terms of comparing those variances. And that's in line with the fact that there are other approaches that have been published in the literature. These have been based on the F test or Levine's test. Next slide, please. So a design that potentially helps with these problems is the replicated crossover design. So maybe we can now look at some examples of replicated crossover studies to explore treatment response variability for acute responses. And I was very lucky to be involved in these studies that you see here. These were undertaken with my friends at Loughborough University. And these were on the topic of acute responses of appetite-related outcomes. The top study was on the acute responses to exercise, and the second study was on the acute responses to a meal. Next slide, please. So first we have to think a little about condition sequences, because we have, at least in the Loughborough studies, we have 4 trials to sequence in a random order for each participant. That's 2 intervention conditions and 2 control conditions. We did this in the Loughborough studies using the sealedenvelope.com facility for randomization. On the right, you can also see a sequence which appears in a study by Steven-Sen and his colleagues. And they also detailed a replicated crossover trial where there are different sequences of 3 of the 4 trials for each person. You can see that which 3 of the 4 conditions actually varies between subjects. This does work in being able to derive a subject by treatment interaction term, but there might be a reduction in power versus sequencing all 4 of the trials. However, to my knowledge, specific power simulations have not been undertaken as yet in this context of replicated crossovers. Next slide, please. So one way to explore the data is to look at the correlation between the 2 replicates. So that's the correlation between the first control group adjusted response and the second control group adjusted response you got in this replicated crossover. So here we have the correlation of the control group adjusted responses between the first and second replicates. This was actually, the data that you see here was actually the peptide YY responses. They tend to be increased with exercise. But there did seem to be an indication that individuals were relatively consistent in their control group adjusted response. Next slide, please. Conversely, here we have an example of inconsistency in response, I suppose. This was a replicated crossover study my colleague at Teesside, Paul Chesterton, undertook on acute responses to a lower back manipulation. And the outcomes were all to do with these Nordic hamstring activity or exercises. And you can see there that there's, especially for the right side, the right leg, you've got a pretty much a flat line across the replicates, suggesting that individuals weren't very consistent at all in their individual responses. And what you have at play there is more prevalent measurement error. Next slide, please. Because we now have replicated conditions in the study design, we can quantify this elusive subject by treatment interaction term, and we can even test its significance if we like. So you can see here that there's general good agreement between the standard deviation for individual responses from the equation that can be used and the standard deviation for individual responses that was derived from the model that we set up in SAS. Like in Alan's presentation, this involved the proc mixed command, so this mixed linear modeling approach. Next slide, please. We could also plot the data like this, and I think this is similar to a way that Rodrigo presented some data he had yesterday. You can see that we have components of within subject variability in response, or between replicates, and between subjects variability in response. And these two components can lead to different scenarios, I suppose. For example, there might be good consistency between replicates, but not necessarily any clinically important differences between subjects. In much of the appetite work that I did with my colleagues at Loughborough, we seem to see individual differences in responses, but every subject seemed to still be a responder relative to the clinically important threshold that we set. Next slide, please. Alan has formulated some SAS code which attempts to quantify these two components of variability that we can see in a replicated crossover. And we hope to present the ideas and behind the code and the code itself, among some other additional things we will cover in a future paper. We're in the early stages of preparing this, but hopefully it could be destined to be the third paper on the topic of response heterogeneity that we've published in Experimental Physiology. Next slide, please. Just as an aside, I just want to quickly point out that there is a relationship between the replicated crossover approach and so-called N of 1 trials, which are becoming increasingly popular, again, in the context of individual differences in response. The latter designs tend to be a bit more flexible, but they still fundamentally involve replication of conditions for one or a number of participants. And there might be more than 2 replicates, there might be 3 or even 4 replicates for a given patient in these sorts of designs. Next slide, please. So just before I finish, I thought I'd briefly add a note of caution. when attempting to identify predictors of response. So, because I suppose the question can be asked, like, if we have to do all this to actually confirm that there's response heterogeneity present, you know, how come that we can still maybe see predictors of response in our studies? A favorite predictor of response to explore is sometimes the initial or baseline value of the outcome itself. And here we can see that as the baseline value increases, so does the reduction of ghrelin due to exercise. That also seems to get bigger, that reduction. Next slide, please. However, I think it was Rodrigo again who also mentioned yesterday that such a relationship might be due to regression to the mean. So please bear in mind that the relationship that I showed you before in here, in this slide, is actually the control condition data where no intervention whatsoever was introduced at all. So what you get is the old fours of regression to the mean and mathematical coupling to make this artifact appear, really. And bear in mind that it might not just be the baseline values of the outcome that where this appears. It might also be an issue when any other baseline predictor is correlated to the change in the outcome, when that predictor is also correlated to the baseline of the outcome itself. So you've got some quite complicated, spurious correlations that might be occurring there. Next slide, please. So just arriving at my take-home messages, quantifying response heterogeneity is at least difficult from a conventional crossover study. And we think that only replication of the conditions themselves can isolate fully this elusive subject by treatment interaction variance and get anywhere near to identifying responders. We think that it's best that replication should be at the whole trial level or the whole condition level, rather than repeated measurements within a trial, which would lead to different inferences. And of course, this approach works for acute responses the best. I hope I've shown you that graphical approaches can help just as much as the modeling. And in the modeling, it's definition of a clinically important change is probably paramount, as it is in a lot of statistics. And just to say that there are links between the replicated crossover trial approach and N of 1 trials. So in the next slide, I'd just like to thank my colleagues, especially Alan and David Stencil at Loughborough, who was very kind to involve me in the studies at Loughborough, and the very hard-working PhD students and postdoc workers who do these replicated crossover studies. They are not easy and take a long time. So we have Phil Williamson, Lorenzo Lolly, Paul Chesterton, Fernando Goltz, and Alice Thackeray, who did all the proper hard work. And in the next slide, I'll just leave you with my final comment. Thank you very much.
Thank you very much, Greg. That's fantastic. And I agree. Keep repeating the talk. But that brings us to the end of the presentations and to the start of the Q&A session. Just before we start the Q&A session, I'd like to just make one or two very basic points. I'm more an enthusiastic amateur than a professional in terms of statistics. And what I really like are pictures, and I'm glad to see that Greg supported that. drawing diagrams and waving your hands about is very useful when it comes to explaining things to people who aren't familiar with statistical concepts and statistical methods. And it can get over concepts very much more easily. I did have one or two very simple slides to show people to begin with. And I don't know if Rosie can do those for us just now. Could just look at the first slide that I suggested was that we should always start off very simply, using very simple stats, taking good samples with lots of values if possible, and then explore them. And exploring means really just drawing diagrams and looking at different ways of expressing them. I'm amazed how many people jump straight into working out means and standard deviations. Medians and quartiles are very much more useful when you don't quite know what you're going to be coming up with. They're much more resistant to being fooled than the mean and the standard deviation. Simple plots like the stem and leaf plot show you the data. And in many circumstances, particularly when we're using derived values like percentage change, transforms of data can be helpful in pulling patterns out of what seemed to be an awful scatter of plots. And then when we test, we should probably be using more robust procedures, particularly when we've got small samples. Robust procedures are much more trustworthy than some of the samples, some of the procedures that are based on the, let's say, classical statistical procedures, which rely on using large samples to estimate populations accurately. So those are just some sort of fundamental points to start with. Can we now start with maybe some of the questions that we looked at that we didn't look at yesterday? I'm just going to bring those up on my screen. I think I've got them here. Oh yes, I made that point actually yesterday, that using robust measures of median and quartile would be useful for some of the data. We have a question: what statistical methods exist to investigate correlations between baseline values and changes without falling into this statistical problem that Craig mentioned of artificial links between baseline values, mathematical linking? Are there any methods to use to avoid the regression to the mean, and would mixed models or growth models work for that sort of concern? What do you think, Greg?
I think I've seen Stephen Sen joke about this, that I think he actually did his PhD thesis on this problem of whether you can correct for regression to the mean in looking at the baseline effects on change. And I think he jokes that he got to the end of his PhD and the answer was no, you can't. The design sort of solution is to always have a control group, and the analysis that would be linked to that would be a baseline by treatment group interaction term. I think that would be correct, would it, Alan?
Yes, indeed. I should have mentioned that in my talk, actually, that if you want to try to explore heterogeneity and attempt to explain it, if you observe it, that a good starting place is putative effect modifiers, like the baseline value. Do people with low or high values get a bigger treatment effect? Sex, age, for example. Compliance with the treatment is another one, and interactions of those subgroup variables with the treatment variable is the way forward. And then you can pull out the difference in the treatment effect for different levels of baseline, or for men versus women, or for whatever it is that you're looking at that might explain the heterogeneity. So rather than looking in a single group at the correlation between the baseline and the change scores, run an interaction with treatment in a randomized controlled trial, and then you'll be able to pull all of those things out. And to the extent that that variable does explain some heterogeneity, you should see a reduction in the individual response variance that you calculate from the model.
Yes, and that actually sort of covers one of the other questions we had from yesterday, which was, do you have a way to do two-way repeated measures ANOVA in non-normal data? I mean, the linear modeling is, again, I guess, doing the same sort of thing, isn't it? There are non-normal approaches to an analysis of variance, but a linear model does the same thing. Is that not right?
That's yeah. Yeah. And I also should have mentioned that I mentioned that it was stripped back code that I ran for my analysis, just as an example. But looking at your simple stats and examination of some of the assumptions that underpin the models, we would always generate the model residuals from the outcome solution and inspect those. And from a visual inspection of the model residuals, you can examine the assumptions that underpin the model, such as normality of the residuals, constant variance of the residuals, or homoscedasticity. You can look for non-linear effects, which often show up as like an inverted U in the residuals, and so on. So, of course, you should always inspect the residuals and other data exploration techniques, because they may point to a need to re-specify the model, as you've said, Gordon, with things like a log transformation, which is commonly used in
right.
human data, which is a normalizing and variance stabilizing transformation.
Yeah. So we have another question on Bayesian analysis, which we did raise its head yesterday, and we should maybe think a bit more about today. I put together another slide of mine, which covered a very good article, but rather heavy one. So not that one. The next, could we have see the next slide? Yes, here we are. From psychiatrists and psychologists are very good at statistics, and that's a very good article, but it's far from simple. The first one I've got there. The second one is a sort of really very sort of primary guide to some Bayesian approaches to testing for differences, and just more generally to point out to people that are watching, that article is from a whole series of articles that I wrote with Valor on different statistical approaches for people who really are not familiar with the statistics as a topic. People who are scared by statistics, I suppose. The guidelines were written, I hope, in simple language. What about Bayesian tests for these sort of things of baseline? The prior for a Bayesian test is often taken as no effect or something like that. That's a problem, isn't it? Is that a problem? Can you fit in a degree of variation into a Bayesian prior?
I don't know about Greg, but I've got no experience of Bayesian modelling with heterogeneity of treatment effect analyses. I'm familiar with them for mean effects, of course, where you combine a prior with the likelihood from the data and get a posterior distribution. But I'm not familiar with their application in this area. That's for quantifying components of variation. That's not to say those methods don't exist or haven't been applied. I'm not familiar with them. Greg, do you?
Okay.
Yeah, I've no experience as well, and I think I'm almost relieved to say that I'd say the major take-home message from both our presentation is design. Design is paramount, and I'd be really interested in if anyone can apply a Bayesian approach specifically to get over this problem of, at least in a replicated crossover, where you can't get to the subject by treatment interaction term from a typical crossover. You know, how that is rectified with any approach. So in the analysis that I showed you on that dummy data in my presentation, I also applied like other popular things like variance components analysis, linear mixed modeling, and the linear mixed modeling just comes out with an error because you're trying to separate two sources of variance that aren't there. You can't separate them.
Right.
So,
Okay.
I'm pessimistic. I think that for that specific problem of getting to that subject by treatment interaction, that it might be a kind of general problem independent of whatever analysis approach you adopt.
Yes, there's another point that's been raised here in the questions that are sort of the same sort of almost imponderables of mentioning here a question of test, retest, reliability of a response. This brings in the question of how replicable an analysis, an assay could be. And this, of course, has its own variance or variability, which is difficult to estimate sometimes. I'm in my field particularly interested in comparing measurements made by different devices, which leads us into sort of the Bland and Altman sort of measurement, where the sources of variance come from the within subject and between subject contributions to the overall variation in a measurement. So I'm not sure that we can answer this question of, if you already have data on the test, retest, reliability of a response, can you account for this? I think it's you've still got the same problem of trying to pick these components of variation apart.
One consideration might be the time between the test and the retest. So a typical test-retest reliability study tends to be a few days between those two, whereas if we're talking about trials and parallel arm trials, maybe with exercise training, for example, that could go on for 6 to 9 months. So there's a question of whether the test-retest data that you might derive from a typical reliability study is kind of time-based, parity-wise, to the trial that you're trying to apply that variability to.
Yes, yes, right.
Some people in the past have attempted to use data from a reliability study in the absence of having a control group in their intervention study. So they might have a single group that they apply an intervention to, and then to try to get at response heterogeneity, they invoke reliability data from another study, test-retest reliability data. and try to use that as the estimate of random error that they're going to subtract from the response variability in the intervention group. And as Greg says, quite often in those cases, the reliability data that's used is very short-term reliability. Whereas if you're going to do that, you would need reliability data over the same time course as the intervention that you're studying. And there's not many retest reliability studies out there that are done over 3.
Yeah, that actually echoes a question that's just been asked when conducting a population study to assess measurements or effects of exposure. Does repeated sample selection from the same study population and estimation of confidence interval take care of the individual variability in that measurement? I guess it would to some extent if you had a homogeneously known population that you knew the homogeneity or the variance in. But that sounds like having a set of prisoners or something you could do experiments on.
Yeah.
I'm just looking to see if there are any more questions. I think, oh no, we've got another anonymous question here. When I use a linear mixed model, should I draw a figure with predicted data instead of raw data? I would have said both actually, but what do you think?
I wonder if that's for exploration of the residuals that Alan mentioned, where you might do a predictive residuals plot.
Yes.
I hope I'm on the right tracks there.
I'm just reading the question now, and it seems to be the fit of residuals against fitted values or predicted values that you typically look at to explore the residuals. So yes, I agree. We're all for graphical approaches to help understand the data from a data exploration perspective, but also from to examine the underlying assumptions of the model.
And the second part of this question is, does the linear model method have a post hoc test? I'm not sure if that's true or not, or possible.
So I guess both examples that Alan and I covered have been the simple case of a two group or a 2 condition scenario.
Yeah. So you don't need it.
You're talking about more than one treatment group.
Yes, yes.
You would think it would be a plan if you're comparing in a parallel arm trial, Alan. What do you think? You'd be doing planned comparisons, wouldn't you, to the control group?
Yes. I'm just looking at the question that the person asking the question says they've got 2 conditions and 7 measurement time points. So hence the question of the post hoc test for the time variable, I assume.
All right.
So yeah, yes. I mean, in principle, all the full suite of follow-up tests would be available to you, depending on what your research question was.
Okay, I think we seem to have run out of questions, and we're getting close to the time we should close. Does anybody else have any more questions, or shall we just draw the thing to a close, Mike?
Gordon, I think there's a question there on trainability.
All right.
Maybe it's not popped up on your screen yet.
Oh, yes. We have here a question. To test for trainability, it would be needed either a repeated testing approach over time or repeated crossover. Let's say you do observe trainability in one intervention, but not in the other for the same people. Could you affirm one approach is superior to the other? How could these be compared? Goodness me, that's out of my pay scale. Alan?
I'm struggling for a definition of trainability there. So I've seen the word trainability used in relation to this responder counting thing that I covered. You know, how many people in a sample do their responses exceed a given threshold that's been set.
Right. Yeah.
If that's what is meant by trainability. I've heard it called responsiveness as well, but it's essentially this responder counting approach.
Right.
You're comparing responder counts relative to a threshold between groups or between conditions.
Yeah.
Yes, you'd have to have some type of threshold in order to make that call, wouldn't you?
Yes.
It may, Gordon, relate to a problem we often have when we're deciding on experimental designs, whether to have two groups or do a group where you have one intervention, then a washout period, and then another intervention, or the alternative. So you're doing a within subject comparison. The problem that always dogs that approach is how long's the washout period and how do you know when it's over? Because we've heard over the last couple of days that, you know, if you really properly want to assess people's, for example, athletic performance, you need to do a psychological assessment as well as a physiological assessment. Now, some of the changes which occur during the first intervention don't go away. And so it almost excludes the repeated measures approach with a washout. Is that a fair conclusion?
That's a point I made in my presentation, that we believe that for chronic long-term adaptations, which can be described as relatively permanent, let's say, that there'll always be some residual at some deeper cellular molecular level. So arguably, it could never be fully washed out, that you'll be starting from a different baseline for the next time around. So I mean that's what leads to our preference for independent groups, parallel group trials for long-term adaptations. I know some people hold a different view, but I don't know on what basis you'd make the call that your washout had been adequate.
Yeah.
Right. Could I throw in a curveball here a little bit and say, could we put up my slide 3, which doesn't maybe apply too much to physiology, but it certainly applies to psychology and things like that. Oh, that's the wrong one. My third slide, that's it. Simpson's paradox, the difference between individuals and a population. If you look at a measure or a relationship like the number of mistakes that you make when you type, it's very different in individuals than it is in a population. You can have someone who, like Jack, who is pretty slow and makes lots of mistakes, but he has a relationship between mistakes and speed, which is exactly the opposite to the population's response relationship, which is shown by Jill at the other end, who is a fast and accurate typist. So if you're making multiple measurements in individuals, you may have a confounding factor for a relationship that you're looking for in a population.
And that's a consideration also for meta-analyses, isn't it? That you might get a different relationship from a meta-regression than actually what's occurring on an individual person level. And I think this is why Alan stressed that because of the power requirements of these sorts of variance comparisons, we're probably looking at meta-analytical approaches, but on an individual patient data level.
Yeah.
I think you mentioned that, Alan, didn't you?
Yes.
Yes.
And that involves researchers getting together and collaborating and sharing data.
Yeah.
But in general, for these sorts of issues, you'd be looking to, in your analysis, to respect the data structure. And here you've got repeated measures nested within individuals, ignoring the data structure, and you run into the type of problems that you've described.
Okay.
Gordon, we've had some questions privately from the panelists to do
Right.
with the size of the N required, which of course always frightens anybody.
Yes.
I obviously work with Alan on experimental physiology, and he often comes back with the comment that this should be regarded as a pilot study because of the number of people tested. So one of the questions is, the prospect of designing a physiological study to address all the concerns raised seems daunting and expensive. Can the complexity and cost be addressed by including the Bayesian approach or statistics mentioned yesterday? Or is there any other, any other way of getting this so that what are currently always having to be regarded as pilot studies can be regarded as not so?
I echo some of Greg's earlier comments that I don't think that a different statistics approach or a method of or school of inference per se, you've still got an issue of relative imprecision of estimating variance components with relatively small sample sizes. And the data that I showed, with 100 in each group was small for variance components as well. So I think the answer lies in standardized design and methods of data collection, and then data pooling exercises, including individual patient data meta-analyses to help get over this issue. I mean, you could have a number of groups around the world interested in the same issue of heterogeneity of intervention effect for some particular outcome, and all following essentially the same design and methods. And it would be a feasible trial to run for each individual group. But they couldn't do such a trial with 800 or 1,000 people, for example. But pooling data using an individual patient data approach might be a way forward. What do you think, Greg?
I think it's a problem that's not going to go away, isn't it? So even if you ignored a stage where you're trying to quantify heterogeneity of response itself, say if you ignored that stage and went on to trying to identify predictors of response, if you did that properly, but in a parallel RCT with proper subgroup analyses involving subgroups in both the treatment arm and the control arm, again, the sample sizes would be large. Even at that point of trying to predict response.
Oh, it's exactly the same thing. So if we talked about baseline earlier. If you wanted to look at the interaction of treatment with baseline or treatment with sex, for example, are there differences in the effect between men and women? You need at least 4 times the sample size as for the original mean comparison of intervention versus control. It's known as the rule of 4. The rule of 4 holds if the interaction that you're looking for is of the same magnitude as the mean effect that you went after, the targeted effect that you used to power the study in the first place. So for any subgroup study, you need around 4 times the sample size as for the original comparison of means. Now, given that in most fields of inquiry, even the comparison of the means in the intervention and the control group is underpowered, as you said, Mike, then it's even worse when you start looking at subgroup interactions and individual responses and so on. So my feeling is that we're not going to get at this issue in single studies. I think we need to pool studies in order to get close to it. And now we're seeing a proliferation of meta-analyses of individual response variants, aren't we, Greg, in a variety of fields, from psychology through physiology and medicine and so on.
Yeah.
Yeah. I suppose the other question, philosophical question you could ask then is it worthwhile powering some studies to come up with, you know, a red line categorical significance answer, or are you just looking for something that's going to give you a lead to a change? You know, a lot of these studies that you look at and you say, well, they're underpowered, they're not going to give you a categorical p less than 0.05, but is that what you want?
I agree with that entirely, and that's why most subgroup analyses, including individual response variants as an example, should be regarded as purely exploratory. They're not definitive. You can't make definitive inferences, but they might point to like differences between men and women in the treatment effect that would be worthy of further study, for example. Just to give you a little story to illustrate this point. A while back, I'm going back a number of years now, one of the major funders of health research in the UK suggested that they wanted all trial proposals to include subgroup interactions. You know, we want to know whether it's more effective in men than women, or for different ethnic groups, or for different socioeconomic classes, whatever it happens to be. When people started putting in proposals for those subgroup interactions with the necessary sample sizes to power the study, well, what's the answer? The trials are too expensive, right? They just can't run the trials. So that rapidly disappeared and went back to mean effects again, and any subgroup analyses being purely exploratory. And that's how you'll see them described in the biomedical literature for the most part.
It's a bit like the other story about the Lancet rejecting someone's paper unless they did a subgroup analysis. And he said, fine, I'll do that, but I'll also analyze for the star sign of the subjects. And he found that Scorpios were more susceptible to the antihypertensive.
Indeed, yeah. If you dig deep enough, you'll find some significant effects.
Yes, yeah.
One of the things I think we see I see quite commonly is a study that is powered for a one variable. Then you get a number of subjects drop out of the study during the course of it. And then you'll see the conclusions made on variables that the study was never powered for in the first place. That seems a bit of a double whammy.
Yes, this is a phenomenon known as outcome switching, which is quite common, and it's something that pre-registration of protocols and statistical analysis plans, etc., is supposed to solve. You pre-specify your primary outcomes and your analysis plan, and then that's in the public domain. And then in the subsequent report, if you start making a big noise about a different primary outcome because you didn't like the results for the first one, then that sort of prevents that sort of outcome switching. You'll usually see a trial powered on a single primary outcome and everything else described as secondary. And the purist will say that you shouldn't make definitive inferences based on secondary outcome variables. So the pragmatist says you're throwing away a lot of information if you ignore things that are happening on the secondary outcomes. But yes, the phenomenon you describe is regarded, I believe, as outcome switching, which is quite common where there hasn't been a pre-registered protocol and statistical analysis plan that's put in the public domain.
I was going to say that touches on the issue of published protocols, doesn't it? Of which there are relatively few in kind of experimental physiology, aren't there? It's almost like the norm for any clinical trial to publish a protocol and state what your primary outcome is in advance, but maybe that should be mirrored in physiology experiments as well.
Yeah.
Well, I think things are moving in that direction in all fields.
Yeah.
So I hadn't paid these guys to say any of this, but that is a perfect platform and springboard for me to say that that's exactly what experimental physiology are doing now. We've introduced registered reports, which Alan will be aware of, where, as I mentioned yesterday, people are able to submit their protocol and statistics plan in advance of doing the experiment, and it will get a review, and part of that review will be looking at these issues. And if people then stick to that plan in the same way as we've discussed with clinical registered reports, then, you know, that, and by sticking to it, we mean having the same primary variable reported, etc., then the chance of being published is significantly increased. The chance of publication bias is significantly reduced, and I think everyone benefits. But we're very proud at Experimental Physiology to be in the vanguard of introducing that into physiology journals.
Indeed, that's a big advance.
Yeah, it's a great idea. Yeah. We have one. Is the final question here? Oh, I think we've probably answered it actually. It's a question about large sample size and the qualitative approach for variability, better than smaller scale studies. Well, I think we've covered that material completely, actually, what we've said already. So I think it's probably time now to wind up. I'd like to thank Alan and Greg very much. I'd like to thank Mike very much for inviting me to chair this session. I've enjoyed it enormously. I think I've enjoyed the whole conference enormously. And well, thank you very much.
Thanks very much indeed for the invitation.
We're moving on now to the chat rooms.
Now we're moving on to the concluding remarks, unfortunately. Gordon, that's me.
Okay, Mike, off you go.
So thank you very much, Gordon, Greg and Alan, for a really useful and informative session. I'm just going to a few concluding remarks before we do move on to a network session. And just to say up front that please remember that the abstract book is available on the website, and this meeting has been recorded, and that recording will be available on demand soon at the website. And this is possibly, we hope, because of the interest in this, going to lead on to a face-to-face workshop next year, COVID permitting. So this has been a rich and diverse meeting, unified by the exploration of variability. Having examined the danger of summarizing data in many areas, there's an obvious irony in me now summarizing a meeting on variability. So I'm not going to. All I'm going to mention is a few high-level points that I think are worthy of note. I'm sure everybody who has attended the meeting has taken something away from it and will be able to use that information in a positive way. My messages, the ones that I've taken away, include, number one, variability can be a friend or a foe, and you need to understand that as you go into designing experiments. Every meeting should include a statistician. Statistics is a separate and independent discipline that requires a level of expertise that some of us pretend to have, but often we don't. I always remember when I was at the Institute of Naval Medicine in Gosport telling the statistician that if he didn't get involved in doing extreme environmental physiology, I wouldn't get involved in statistics. So we have to appreciate that that is absolutely critical for us to do the correct experiment. And as I've just discussed with Greg and Alan and Gordon, that statistical input is far better to come at the time of a protocol and an experimental design than as part of the review of a paper that where it's really hard to correct errors made at the protocol stage. To fully appreciate the data we have to step outside of the silos that we inhabit, which are artificial silos created by the way we're educated. And one of the things that's come out time and time again in this meeting is the importance of collaboration between different disciplines, and collaboration within the same discipline, just to make sure we get an appropriate number of tests undertaken to meet the statistical requirements for being able to conclude on the work we've undertaken. So they're my take-home messages: statistics and collaboration and variability, the importance of it. I want to finish by thanking our speakers and our chairs. I want to thank the attendees for their interest and their questions. I want to thank the Physiological Society, the European Space Agency, and the Energy Institute for supporting this meeting. I want to particularly thank my co-organizer, Professor Igor Mekjavic, for his contribution and his input into the initiation of this meeting. It was a bold step to have a meeting focusing on variability, but I think the number of attendees and the amount of interest has vindicated that move. And last but not least, I want to thank Rosie Heinard, who works like mad in the background, making sure the presentations are there, you're seeing the right slide, and that everything runs smoothly. Without Rosie, this would definitely not have happened. So thank you very much, Rosie. So now there's an opportunity to go into a networking session. You will find on the chat function a link that will now enable you to choose to go into a network session. We'll all meet at that link, and then Rosie will divide us into breakout rooms for a discussion. And that discussion can involve anything that we've discussed in the last couple of days, the usefulness of the meeting, and where we might go with the workshop next year. So those of you who are going to come to the network session, I look forward to seeing you in a moment. Those of you who are now departing, thank you very much indeed. All the best.